with hydrothermal vent fields and underwater mountings which creates biodiversity hotspots and animal life that you see nowhere else on the planet. This is a mineral-rich area that the Norwegian government wanted to open up for deep-sea mining, but that was stopped at least for the coming 4 years. And while they want to extract minerals here, we want to protect this area. So we're here to document it together with scientists. I'm going to go in and meet one of them in here, in our wet lab, and tell you a little bit about a dive we did yesterday. Over here by the samples, you can see the scientists are working hard with the samples from the ROV here. And one of the samples you can see here, this pink little piece of coral, it's actually called bamboo coral. And we don't quite know exactly what species it is yet, but I'm going to hope we can see a close-up in here. In here behind me, Solvin Sankel is working. He's our blackwater photographer. Hi, Solvin.
Hi, Daniel.
I want to have a look at some of your pictures. Can you show
Sure.
me?
We had basalt rocks and all the animals living on them. And we have here a crinoid. And we also have a sea spider.
Ooh.
And sponges, of course. There are a lot of research going on with sponges. And the famous bamboo coral. Here you can see the polyp.
So this is from our previous dive that was happening on what we believe is a bamboo coral garden. It's creating like these 3D structures stretching over 800 meters over one of the mountain ridges. The scientists believe that this creates home for other animals, and we found lots of eggs there, which indicates that this might be a nursery for Arctic skates, so this ray species living out here. But the next dive is going to be something totally different. We're going down to 2,800 meters at a site no one ever dived before. So we don't really know what to find, but it's going to be exciting for sure. You don't want to miss this. Let's go live.
Today, at the beginning of the day anyway, Sergi and Anna-Helena again. And as you see, we're still on the descent. We are approximately 150 meters above the seafloor, so we do have some time to descend still. All that beautiful light you see in the water is not from the sun, but from the wonderful light that we're bringing down on the ROV to see what's happening down here. We're going to an area we have never ever visited before, and when I say we, I mean we as humans. Nobody have been here before. We are a little bit northeast of an area called Fåvne, which is an active venting field. So we will look at some mounds and some hills and some other interesting bathymetry. So we have just been able to look in the bathymetric maps and decided an area that looked interesting, and we are looking forward to exploring this together with you guys.
Yeah, we're going to be something around 2,800 meters, which is almost 3 times the depth that we surveyed in the previous streaming. So it was like something around 1,000 meters depth. Some cases 900 meters depth in this bamboo coral reef, right?
Bamboo coral garden?
Garden, yeah.
Bamboo coral garden?
Not yet a reef, but
Who
okay.
knows?
So yeah, we're expecting quite different communities, but yeah, it's not going to be until we reach to the bottom that we will be certain about this.
So right now we're sort of just waiting, and this is a little bit later in the day than our last live sent dive. So this morning, both Sergi and I have actually had time to have breakfast. That was
Yeah.
lovely. What did you have?
Toast and some fruit.
Some fruit, and I went for oatmeal. So we're all good and ready to go for a long time in the shack here with you.
Oh,
Ooh,
at that.
look,
That is a
octopus
Dumbo octopus, I think. Could it be?
dancing by. Yes.
Uh-huh.
I hope everybody got that one.
It's a bit elusive.
It was a little bit away from us, and we're going down pretty fast to reach our bottom at 2,800 and a little bit maybe, and we're now already at
Oh, can you see the
2,740.
bottom now? Can you see it now?
No,
Is that? Oh,
no,
no, not yet.
that's just, I think, plankton that is passing by us. It's almost like sitting in a spaceship and
20
flying
meters?
through the stars.
We are 20 meters away from the bottom.
20 meters away from the bottom, according to
So this
the
might be, yeah,
pilot.
right. This is
No, this is a thing on the lens.
here. Can't wait to see what's in there. And yeah, we're going to be climbing, right? A little bit, so it's going to be like a ascent.
We normally want to do a little bit of an ascent. It's easier for the
Here.
pilots to see when we're going uphill than when we're going downhill.
Yeah, those are things. No.
Now
Oh, look
we
at that,
see
look
the
at that, yeah.
seafloor. Wow, look at that.
Yeah.
Landing.
Great.
Ooh, and here we see, I would say, I hope none of my geologist friends are being angry with me, but I think we are seeing some sort of old-ish pillow
Do you
lava.
think, shall we go?
Yes, look
this
at the, well, check it out. You are,
this
there
is
is
an
an
octopus.
octopus.
Yeah.
There is an octopus hanging out and looking at us. Let's see if we can zoom in on it before we zoom in on the
Oh,
seafloor.
wow, 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 look at that.
There is a lot of things in the water
Sorry
as well.
about that. It's not as close as we would expect. The beautiful footage of this super beautiful creature.
And it's swimming away from us.
Yeah. Too light. I mean, too bright for this guy to be in here. Okay. Coming back to what we are also interested in, which is like describing life. Mainly these benthic, these organisms that live at the very bottom, that they live attached to the very bottom, and also some of them that are flying away. As you can see, there's a really strong current, right?
Yeah.
If you see the particles just flying away to the right, you will notice, and you will have an estimation of how strong the currents are in here.
Well, as we said,
Anna,
Yes,
not recording yet.
I am ready to start recording if you are ready.
balance as well.
Oh yeah, we want to do that as well. Maybe we should start with doing, yeah, let's record and then we do white balance.
Okay, can I?
Yes.
Okay, start recording.
So we've started the recording, and then the next thing we should try to do is to fix the white balance so that we have a good color level for you guys.
Oops. the dust is going to be washed away in a sec. You'll see.
Yeah.
Oh, no.
But it's quite a sedimentary bottom we're landing on, and it's interesting because it's sediments in between all of these pillow lava, I think it looks like, or all pillow lava.
So
So there's a little bit of administration right here at the beginning of a dive to get everything ready.
Can you give me the coordinates?
The coordinates.
45.
45, 93, or 98.
93.
95.
3. 5. Longitude. It's
3,
3 already.
52, 17, 7,
7.
4, 6,
4, 6. And depth is
2768.
27.
68.
6. 8. So
Yeah.
almost 2,800 meters right now.
Yeah. That's a good place to hang, I think.
We're just
And
checking that there is a big difference here, but
our pilots are trying to find out where it is a good place to hang. I think this is again a quite wild landscape we're having here. Look at that. So when our pilots find a comfortable place to either hang or settle down, we can fix the white balance, and then we're ready to rock and roll, I guess. What do you think, guys? Do you want to just hang here to do the white balance?
Yeah, second, yeah, we'll just try to get it's
Yeah.
not obviously level here, so. Right here we can go.
Beautiful stuff. Sorry.
Yeah.
Can you
Potentially in the meantime, right? Just have a look at some of the creatures that we see in front of us.
Is this another sponge, you think?
Yeah, indeed.
I think that
It's one of the unshaped ones. Oh, sorry. It's time for white balance.
yes, we're ready for white balance.
we'll keep the oil there.
And I think we are seeing, is that also encrusting sponges there on the side?
Yeah, we may have a look.
Yeah, afterwards.
Different sizes,
Yeah.
different colors, but still sponges dominate this area.
Oh, this is a rocky, deep sea area. And we are getting ready to set the white balance of the video so that things do not appear too green or too yellow or something like that. And for this we bring a tool. It's not only used for this but also used for this, to have a known white surface that we can calibrate against.
To take this out,
You can come up, yeah, it's quite tight. It's
you want to
And look, there
What is
is
that?
a fish.
Oh, yeah.
you kind of like.
He's swimming with his head down and his tail up. Well, that's a beautiful way of swimming.
Hopefully we'll get some other fish like this during the transit, so that we can just spend a bit more on the beauties.
Yes, because
segment.
today's transect is all about looking and exploring, and we are not doing any strict mapping like we did on the previous
Bring it all in.
live.
So shall I stay here?
You shall
Yeah, so you just zoom in a bit and try and get more
Yeah.
white on the screen.
Yeah.
zoom in.
You tell me when
Now.
you're ready. Now?
Yay, zoom in.
Close it, alright.
We are zooming in on the white to make sure that we get as much.
Oops.
Yeah, oops.
No. There's something wrong there. Since
There is good.
Second.
it goes all the way down.
Yeah.
Right, yeah.
Our zoom is a little bit
think if,
finicky this morning. But we will see if we can fix that.
Yeah, go ahead with this.
So that we can make you the most smooth and beautiful video we can.
Let's check your settings out.
Let's just take those away to make better
You wanna
workplace
sit-
for you.
sit here?
That's alright, alright.
I'm always impressed with how it's possible to repair something that is so far away from us, from this box. This is long distance fixing.
Channel 4.
Highness is always the connection, so we'll leave it like that for now.
Let's see this. What's that?
And then you can make it says for 2 or 3 minutes and then back again. We try again. It can for some seconds and drop. So just in case there is any short circuit, it will not exist.
It stays in one, right? Instead.
Yeah, it just presets to that, yeah. So this one, I might have to power this one off just now. Maybe it's because this one's powered up as well. Should be a little thing that says box or apps or
Yep.
uh,
Do you want to sit here?
no, it's, I'm just trying to find it with the light.
Yeah.
Should be a power one in somewhere. That's in here.
We can turn on this light. Everything
Okay.
will be a lot easier.
Just need to find this one. Turn it off. That's in here. That's it, right. And then I think it connects when that's powered up as well, so.
So we are still just trying to fix the zoom on the camera so that we can
It's all right. Yep.
smoothly zoom in on all of the beautiful animals that we are going to see, hopefully. And while we're doing that, we're seeing a beautiful red jellyfish just passing by, floating by.
Once this powers back up, it might do it like, you can do it on the software here as well, but it seems to be picking up.
Do we need to push something on this side?
No, that's okay,
Yeah?
Should be. Sounds PC to.
So maybe I should put this light down here again.
Try powering this one back on against this first one.
This?
Yeah.
Here?
Yeah.
Yeah.
There.
There's a lot of things happening in the shack right here. Now we're still trying to get the camera to respond to the zoom.
Yes, yeah.
Yeah.
But the zoom is not responding. So we are trying to fix, so this is the arm moving and not the zoom moving. We are trying to fix the zoom because of course we want to be able to zoom in on all of those
just seeing if it was
beautiful
stable,
animals.
and then after 3 or 4 minutes
I guess the only thing we can do is just have a look around.
just
Yeah, I was
Yeah.
wondering if what we are seeing there on top of the left hand
Left.
clump is a sea star,
that's
in addition
yeah.
to all of the
At the very top, right?
sponges. Yes, because since we have so many sponges, it would be nice to see a sea star as well.
Show here or not? No zoom yet. Yes, J2
we have to see when we start moving what's the line that we are doing here.
I can tell you that we're hanging out in addition to being at 2,767 meters depth, it's also relatively cold. It is minus 0. Almost 7, 0.68 Celsius down here. And this of course is possible because the water has salt in it, so it does not freeze at 0. It freezes at approximately minus 1.7. So we're 1 degree away from freezing point in this
I'll just
water.
get the white balance if it does come in there, and I'll just hit it. Just rotate a wee bit, clockwise, yep. That way,
We're trying to set
clockwise a
the
bit now.
white balance
Oh,
without the zoom.
Rotate clock, yeah. Bit more, bit more. Rotate a bit more, yep. That's done by there.
To see if we can at least get that going. But it seems right now that our camera box is not really responding, so we are trying to fix that.
Can be fixed. So it means that we cannot do like detailed shots. We cannot take detailed shots of the specimens that we're gonna be watching during this transit. But
Yes, we're still able to move the camera up and down and to the side, but we're not able to zoom right now, but that will come hopefully. And because we have such a great gang of pilots with us, they know how to fix things that are almost 3,000 meters
yeah.
deep. Oh, look at that.
it's still like
We have a
Still,
zoom.
responding, right?
Still sticking. So you can do it on the software here, but it's it's not going to be as nice zooming on the software because you have to use the mouse as opposed to the box.
Yeah.
But I think the box is it's maybe like a bit of the connection here is maybe dirty or something.
Means that it's not on focus right now, right? So it's gonna.
Yeah, it should.
Yes, there's autofocus
Oh, yeah.
again.
Oh,
That's good. And will it respond to the white balance trigger?
Well, yeah, but it's not quite set up yet. I think my mouse is gone now. Fix that, lost the mouse. Just kind of
So while we're having this beautiful view of our white balance measurement, the white balance circle, maybe I can ask some of you other guys who are not helping fixing the zoom, why do we have the metal plate in the back? Is that for scraping off things when we're sampling?
What do you mean in this?
The metal plate that is in the back, is that for some reason, or is it to make it stronger?
That's just so it stands up straight. 'cause
Ah,
it's round.
it's round and it would tip over. So this is a balancing thing. There is a lot of engineering that is on an ROV that we never think about as non-ROV people. Of course, it's important for the hand to be able to get to the manipulator to get to that holding place on the round plate here.
This camera, right? It potentially have an idea of where the arm is sitting so that
Yes.
collected us. Yeah.
Yes, so this phone camera use that for all minutes. So it's where they're going away.
Yeah.
Make sure it's up and it's
We
loose
still
now.
managed to get the camera right. Nice that it doesn't work, so we can steal the camera.
No?
That was maybe. So,
Now that we
which, that's great. Great if you place it in the ground.
Do you want light?
Ah, it's okay. Box. Yeah, it's a USB extender somewhere. Keyboard, mouse. Disconnected. It's locked up.
What you don't see from your possible view of the shack is how this entire area is just filled with computer screens that are hanging on the wall in 3 heights and how many? 1, 2, 3, 4, 5, 6 to the side. So we have 18 computer screens here, no, 16 computer screens here. And all of them are doing different things for us. So we're trying to connect a computer mouse to the right screen, which is some 16 possibilities. So there is a lot of electronics to find out the right paths to here.
Someone's maybe put the laptop down.
Ah,
It's disturbed the.
should we move some of this to find
so
the
it's
laptop?
back in there. I'll have a look at the other side now.
The space is also utilized to its utmost down here, so there is not a lot of space to move around things.
Yeah, really.
Making it more difficult to find the right port.
It's not working right.
Is it hanging?
No, it's not responding.
Yeah, so it's just getting stuck in.
Yes.
Yeah.
So if you click once, move it forward, then it goes forward.
if you are
All the
able
way.
to do the white balance or not?
Let's try to do the white balance. We zoom in all the way.
Yeah.
Well, when we let go, it still zooms in all the way. And let's see, so we are on the channel 2 and we just press right.
Okay. you
That
do.
looks good. That looks like a better white balance, not
Yeah.
so orange. And then we can see what happens if we try to zoom out again. No response.
Mouse came back here now.
No response now.
No, but I think we were able to do the white balance. So
Yeah, we've done the white balance,
Yeah.
but now we only see a white field.
No, that's because it.
Oh,
right
there
up,
it
right.
goes back. This is a very either responding or not responding zoom at the moment.
Yeah. Try and keep the best way on boats. Just looking.
put this on narrow view again.
Yeah, let's go on just now. You can carry on just now if you want, and I'll go and see
Yes,
what.
let's go and have a general look at this area without doing too much zooming, but maybe a little bit
I don't want to freeze it because
pointing
the
back
TN
and
is not
forth.
in the right position on the T4.
Okay, we'll open the starboard door.
And we have made a plan for this dive, as we talked about, to go over a couple of small hills
Yeah,
to see
as far as you can because
how
you have to
they
use
differ on
this-
different sides of the hills and
Okay.
what the hills are. We think they look fun on the bathymetry, so we want to have a look and see if it's interesting in real life and not only on the bathymetry. Do you want to have this back, maybe?
Sure.
We take this here. Here is your notes. And I'm getting ready with this, so here is your pen. Take your pick.
This one was
Yeah. So sorry for that delay, but that's how it is to work with a large machine that is almost 3 kilometers below us here in the middle of the Norwegian Sea at, how far north are we? 72.5 degrees north.
72.4.
Yeah, so we are quite far north. It's midnight sun. Well, right now it's not midnight, but it was midnight sun this night. And so this, it's always light up above the water, and it's always dark at these depths where we are now. And we will start exploring how this quite dramatic landscape is looking. And again, I think we're seeing some pillow lava that have been sedimented, so this is not very fresh volcanic area, but an area that has had some time to get slightly sedimented. And
Because of all maybe 0.20.
yeah. And as we talked about,
Maybe
but
I'll
not
wait
really
for you when you pass through the boat.
zoomed into, are a couple of
Yeah, that's
sponges, fan-shaped sponge, was it?
A fan-shaped sponge, yeah.
Fan-shaped sponge and some encrusting sponges on the left side, and the fan-shaped sponge on the clump of rock that is to the right?
Yeah.
Yeah.
And there is some actinellids, I think, on the rock back to on the left right now.
And I see
whitish one.
on the very left,
We happy to go? Yeah.
Yeah.
on the very left
So we're
rock,
gonna start
there
a
is a
transect.
shrimp. Let's start a transect. And from now on, we move along in our unexplored area.
green. We will turn.
And you can see there is a lot of moving things in this water, so that's not only sediment but also planktonic and slightly swimming small animals in that water. And let's now see if we can find more fish or maybe even another octopus. Right now I think we're seeing small crustaceans and maybe some
This is the direction we need to take?
maybe some marine snow or all of that sediment coming in very, very small packages falling down to the seafloor. So the plan is to move slowly along and have a round. Like
It's gonna be up this mountain.
like a bumbly
Look
walk.
at that. Yeah, you can see them.
The
all
rock
the way
formations
through.
here are crazy.
Yeah, they have this fun shape. I remember we collected one of those, one of the previous times. The one with brownish at the very center, and then the edges being white or pallid.
Is jumping a little bit.
So we're still trying to position the ROV in a way that we see what's going on, because we're going to be moving upwards in this mountain. Again, nothing to worry about these sedimentary plumes, because they're going to be washed away thanks to the currents in a while. This is a result of the ROV
Lifting up
lifting and
after,
starting to swim.
yeah. Fly.
Yeah. Flying, yeah.
I also think about a little bit of swimming, but we're flying. Underwater flying.
We have no reference on here.
Yeah.
We have no reference on that at the moment, if you're on the line or not.
Guys, can you reply to
We have plunger again.
Yeah. Yeah, can you, they just call this the dry lab?
out the fisherman fisheye, please?
Right. Do you want to answer them? The dry lab?
What he wants.
The dry
He
lab's
wants-
calling us.
He wants to have
The
the
dry
Yeah,
lab,
right, right.
Because we are now trying to put the ROV in the right configuration.
Yes.
If you could talk with the dry lab.
Yes.
Yeah,
So
It'd be better. My
we need to wait a little bit.
yes, we will wait with the fisheye, and then we call. After we have positioned the ROV, we can play with other cameras.
We're going on again.
Yes, but go down.
Thank you.
Go down. I've got 3 times on that.
So as always, in the beginning of a dive, there will be a lot of administration also for the pilots. And at the same time, we're trying to fix this zoom thing.
No, mate.
Yeah, no, we just come
So what's the zoom, is it?
Zoom, mate, sticking again.
Okay, you power cycle is it?
Did I?
And she's fine there.
It's fine now.
Still coming in like it was sticking in there, so I tried getting up there.
No, no,
Code
so
4.
see the steer up. So you gotta take that out, connect that to that.
This working together.
Right,
So it's-
that's what it's going on.
Yes.
That's what it is.
Yeah. So do you want to go on that now or what?
It's going to be a bit more difficult for the others.
It's just some most, yeah. I'll hang around for a while.
I will switch you over. Switch you over just in case, because I was stuck in that way.
So we're doing another attempt to repair the zoom.
So that can be as labelled as now.
Huh?
So that can be the mouse is labelled as now. Must be for that purpose.
Which I think has included waking up the boss too early for his shift.
Yeah. So we go 45. So I could just start moving ahead and see if it's sort of way advanced. Just jumping.
So what we can see is that, as we talked about, there are patches of very fine sediment in between this old volcanic rock area.
Find it out.
and there are some sponges sitting on the different rocks that are sticking out of the very fine sediment. And of course what we don't see are all those animals that are living inside that extremely fine sediment. Why am I saying that I think this is extremely fine? It is because of the color and because of how those clouds form when we are landing and taking off. They're really, really finely sedimented clouds. So our pilots are trying to line up the ROV in the direction of our planned dive. We will try to go approximately northeast. Yeah. So we're lining up for that. And then in parallel, whoa, that is a very beautiful ctenophore. Swimming in front of us there. We're also trying to fix the zoom so that we can give you a more interesting experience along our drive. We also fly. We also want to, of course, see what we are flying over. But Sergi, do you think this is again a sponge?
That one that we already collected in one of our dives. I'm gonna ask you to see whether is there any idea.
Yeah.
Identification.
Julio is sitting in the lab and following us, and will take over in some time when we have a shift change here. But first we are going to work a little bit with you. Ooh, now if you look at that sedimentary field, you see all those little holes or pop marks. They are for sure where animals are living. Either worms or clams or somebody.
Lollipop sponges.
Or maybe the lollipop sponges are attached down there. Do you think they can retract? That can also be. How about the cereantid, the anemone that can pull down into their own tube? They also will like to sit in those, and they will hide when we get close, and they get stressed.
Yeah, it's underneath.
And if I remember from, I can take away those papers.
Is the keyboard sticking?
It's back there. If I remember my understanding of sponges, this vase would be Brasilida?
Yeah, I think so. Those are exactly aliens.
Yes.
But not the same species that we saw in space, right?
No, it looks different. It's a lot more brown on the outside. Is that because it has been taking in all of this slightly sedimented water, maybe?
Okay, give it a second.
Yeah.
And we see that big opening where the water will come out at the top there. But as Sergi said, these are glass sponges. So just to remind you, they are the animals that are made of glass. So they are fragile, but very beautiful. So all of their spicules or some of their spicules?
Yeah.
All?
We have a species, yeah.
All of the spicules are made of glass.
Yeah, those almost like things, yes, of
Yes,
course. There is
of the
another
glass
group
sponges.
of sponges. They're called Calcarean sponges, and of course in this case there's no the same kind or similar kind of skeleton. So I think we're about to fix it.
Yeah,
Hopefully.
that will be wonderful. And What do you think those small white dots are?
Not entirely sure.
But maybe they are also small white round sponges?
Yeah.
Small
yeah,
white.
probably, yeah.
We call them the small white round sponges for now, and then when we have the soon back, we can examine them better.
There we can show, but it doesn't seem to be moving kind of on the books.
So I think we're coming to a quite steep uphill of this sediment and rocks. So we're back into that very dramatic landscape.
Very steep, you see?
Yeah.
And you can notice from
Inches. Just for a moment. That's why I couldn't get it. I was only going for. I paid for.
you get the motion that you see.
And we're having those little round white sponges, maybe, Sergi?
Mm-hmm. Yeah. We collected one in the previous dive.
Yeah, come on, slowly.
Ooh, look, it's almost like those rocks, those volcanic rocks are just hanging in the sediment here. It looks crazy.
Okay, we would
And
like to
there
have
is a shrimp
direction
having a
045
right
at
down.
0.1 knots continuous.
So the shrimps seems that they like to sit in this
Okay, let's see, thanks.
very steep sediment with these outcrops of volcanic rock. And then those fish. About where they're swimming, they're mainly floating with the water. They can do a little bit of control, but not super much. So when they seem to come crashing into us, it's mainly because they're maybe just hanging there and we are slightly crashing into them.
Some crushed sponges.
And some anemone, yay. We should start recording some of these again now. So. And is that the pink thing is also interesting sponge?
Yeah, the white thing just below was an interesting sponge, and there's two of these hexactinelids. Probably not the same species that we've seen so far in our previous dives.
Thank you, Paddy.
Is it fixed now? Yeah, thank you so much.
Thank you.
Yeah.
So,
Good to go, yeah.
out of his sleeping time came Paddy to fix everything with the Zoom for us. And hopefully from now we will have an interesting dive, and when we are getting a little bit more control, now we can again look at all of those
I'm pretty
holes.
sure those are lollipop sponges, you see?
Yeah.
I just try to hit you from the shadow, like this. Just have a look.
Those holes?
Yeah, the holes I don't know. But they were some lollipop sponges, yeah.
Yeah, there are some lollipop sponges here, and yesterday we decided we would call them Stylocordula.
Stylocordula, yeah.
And then I think some of those other holes are
Yeah, I see.
maybe worms or maybe something else. I think that is a worm sticking out actually.
Yeah. Okay, so let's just...
Maybe a sabellid worm or something like that. They like to
So
stick
you
out.
have here again this huge hectinelite on the left hand side, right hand side. And then is that a... See, this is a crusting sponge. No, not
And
that
an
sponge.
anemone on the side of that rock below. Now we are talking about where we are looking first when we are where is our focus when we are looking. If you are a sponge expert, you will of course always look first at the sponges.
Can we increase
The
the
most
speed to
conspicuous
0.2?
organisms.
But
And this
it's-
is, yeah. And this is a hexactinelite that we didn't see the other day. And those things, this spaghetti, can
The
you-
spaghetti?
this is white spaghetti like stuff. Those are sponges too. And this is a really common species called
Lisodendrix complicata.
We will never forget this name.
I am trying to learn Latin names of sponges here, so it's very good to sit together with Sergi because he knows them already and can help us
A
learning
few.
them. And then I have a cheat sheet in front of me as well, so I can at least say some names that are standing there. And we are flying. I think maybe if you are when it's good for you, we might be able to take out that other camera, that the sextant, yes,
Okay.
when it works for you.
You see, this is another fun-shaped sponge right here.
Yes.
It's a bit difficult right now because it's super steep. Oh no, it's not a fun-shaped sponge.
Looks like one of those balls.
Coming out.
I can take those until we have something to note.
Yeah.
And then you have a little bit extra space.
Yeah, thank you so much.
It's really a little bit like Tetris to find the space for all of the things we need to have here. So today there's been quite a lot of
So you see,
technicalities in the beginning, but I think now.
Sextant is fully out.
yeah,
Thank you so much.
this thing that just appeared at the very bottom of the image on the left-hand side is a special camera. That is
That
kind of like
maybe-
a 180 or something
Yeah,
like that.
so you might
Why?
already see the film from it if the guys up in the production are playing. But we are looking at the animals on the back there. I think there is another big white anemone.
Yeah, and the thing you can see now with orangish color is another sponge. It's a fun-shaped sponge, Lisodendrix. So we will be talking about the position of different things that appear in the video, sometimes referring to the two lasers that you see in the at the very center of the image. So I guess you see them.
And we should say that there is 10 centimeters or 100 millimeters between those
You
two
see them now
dots.
at the very sea bottom.
These lasers are going out in parallel to each other from the ROV so that we know always what is 10 centimeters.
Those are little lollipop sponges.
Okay, we have some
On the right-hand side.
Stylocordula.
Yeah. There's a few sponges here. Difficult to make a guess on the species, the only species that we are Well, one of the ones is this one that appears just to the right of the lasers. You can see them at the very bottom of this rock. Those are Lysodendrorix complicata.
And since we're talking so much about this Lysodendrorix complicata, I think it is one of the favorites of Sergi, right?
Yeah.
At least right now, it's one of the favorites.
Yeah, I would say the most interesting for our purposes. So we can talk about our purposes.
Yeah. Why don't you talk a little bit about the Lysodendrorix?
So
the
one of
spaghetti sponge, I call it.
So one of the objectives, scientific objectives of this cruise is collecting organisms that occur in different areas, on the different areas of we're going to be surveying, and also that they occur at different depths, different bathymetrical ranges. So this is super important for us, since we want to identify which are the connectivity patterns in between the different areas that we're going to be surveying, and also at different bathymetrical depths.
So, is one of your theories that it might be different species if they are deep or
not really
or shallow, or if it's just different populations that do not have a lot of contact?
Yeah. That's one of the things that we want to test. Whether they are different species or not, they look alike. We've been collecting samples from up to 2,000 meters and up to 900 meters with the same potential new species. And if they happen to be the same species, we're going to be testing connectivity, but also we're going to be trying to identify patterns of adaptation. So you can imagine that the conditions at around 1,000 meters depth, the pressure conditions, are going to be a bit quite different from the ones that we are getting in here, like almost 3 times the depth that we've been surveying yesterday, for instance.
So you want to see if they are then genetically slightly different because of this.
Yeah.
And I think I learned the other day, you told us that the Lysodendrorix complicata and other Lysodendrorix do not have another of those beautiful red jellyfish.
Yeah, beautiful, yeah.
I love them. They are so peaceful.
You can see now.
And remember, jellyfish are in the same group of animals as anemone and corals. They are all in the Cnidaria, and they are named so because they have stinging cells.
Is there a fish?
There's a fish.
Yeah, indeed. Just ahead.
And I think these are
20.
I think these are Sibel, or are they either Sibelids or the lollipop fish? Lollipop sponge, sorry. But that was a fish. So we were talking about that Lysodendrorix, that it does not have larvae that are floating in the water. They are brooders, so they make their babies, and it's still a sexual reproduction. So there needs to be a mommy sponge and a daddy sponge.
Yeah, but they are hermaphrodites.
So they can help each other.
Yeah.
They can do sperm over to each other's eggs. In a way.
Yeah.
But we need to have 2 sponges close to each other, and then they will breed the babies inside their sponge.
Yeah. So we were taking some pictures yesterday from some of the specimens that we collected, and we happened to find some of these embryos in the tissue of Lysodendrorix complicata. It means that at least some of the individuals in this population were actively, they are like in the right time to be reproducing.
And would you call it pregnant if it is a sponge?
Yeah.
Yeah.
Well,
so we found pregnant sponges.
Yeah. Just no
That's
fish.
cool. Another fish coming up here. But just to finish up with that Lysodendrorix story, one of the things that this implicates is that, they are sabellids, those worms that are sticking up around it. Look at that, that's beautiful. One of the things, when you have a brooding animal that is not having a free living larvae stage, is that they don't spread so much. And this is why I think you want to look at the genetic connection between different populations, because how can we find out how much contact these different populations have, right?
Yeah.
Yeah. That's a very interesting question to look at.
So this makes Leucidonorix complicatus a very good candidate to study these connectivity patterns. I mean, the adults do not move, and the embryos...
The babies don't move that much.
The embryos are going to be producing, they're going to become larvae, and these larvae are going to be released into the water column. And there's not going to be much time until they need to find the right spot. It means that they cannot travel, let's say, that much, so they have a limited dispersal ability. And this, under a population genetics point of view, this is one of the things that me as a marine ecologist doing genetics, doing connectivity, this is one of the best candidates to be studied.
Could we zoom in a little bit on the sabellids?
Yeah, sure.
We are going to play with the zoom since we have it again. So what we see are worms that live in tubes down into this very soft sediment, and then they come out of their tubes with their tentacles and sit there and search for food. And they must be just picking up little
organic matter
that comes down through the water column and land on them.
It's not that they are predators, as we will talk about later with some of the species that hopefully will also be in the video. But yeah, these organisms there seem to be, I don't know, we have to have a closer look.
Yeah.
It could be umbellula-like stuff. Could be?
Too small, I think.
Yeah.
But there is one, it has this
I was getting this
head and then tentacles coming out of the head.
Sometimes the rocks appear, so...
Those aren't they umbellula-like? I think they are cnidarians. So definitely we're gonna need to sample these guys so that we can identify.
Yeah, maybe they are. Do you think that they are not worms, but that they are? It's so far to. It's difficult to see, but stalked crinoids, maybe.
Could be. I mean, there's a bunch of organisms that they have gotten to similar evolutionary solutions for getting the best out of the
energy coming down through the water, huh? Make a stalk so that you get a little bit up from the seafloor and have your feeding area a few centimeters above seafloor. Is that the general strategy?
Yeah,
that's
for some
of
these different phyla, which are big groups of organisms that have evolved independently, but they have in some cases reached to a similar solution in terms of getting the food from the water, from the water column. Yeah, there's a bunch of them.
Maybe there are several different things, but they look quite similar, all of them. At least from this distance, but that was, of course, always a problem.
And there's a shrimp at the very back image.
Yeah.
On left hand right of the image.
And shrimp like to stand on the bottom and just hang out and look after food. Even though they are very good swimmers.
So quite a different habitat if you compare these with our previous dive. And
yeah, if you were with us last time we were diving, that was very different. It was also steep, like this has been at some times now. But
slow down.
But we are going over a very soft sediment here, and as we said, I think there are a lot of life right below the surface here. So I would definitely not call this dead at all. I think this is just hiding a little bit from us. But there are several animals that live right on the surface. They are normally quite tiny. And I have been spending the last 5 hours of this first 5 hours of this morning's shift sitting and looking at tiny little animals in the microscope that we sampled with our last dive, and the average size was 2 to 3 millimeters. And when we see how small 10 centimeters is here, we can understand how it's impossible to see some of those 2 to 3 millimeter animals.
Jellyfish is past.
The jellyfish come in all different types of red and dark brown, and that's really cool. I wonder if what is making them look so different in color, if it's just how they hit our light, or if it is what they have been eating, or I have no idea.
Regerovic. Can we come to the 0.2 knot speed? Okay, thanks.
Okay, some rocks, which means that there's going to be sessile organisms. It's not that there's also sessile organisms in...
There are definitely some of the Stylopadula, by the way.
Yeah, you see that one.
50 meters to the volcano.
Oh, you think? It's, this is potential.
Is that
this is
another
yeah.
Lysodendorix?
Yeah, a Lysodendorix.
So there is a reason we have been talking about this the entire morning so far. They are very common in this area.
Yeah, indeed. Now we are at
2,750 meters.
Yeah, and at some point we're gonna be sampling and trying to get samples from this area. So it's really important that we sample, for our purpose. Otherwise we cannot identify species, otherwise we cannot identify this connectivity that we were just talking about some time ago. And it means that in some cases we're getting whole specimen. In most of the cases we're going to try to avoid this. It's just taking a piece of these specimens so that we can bring them up and we study them under the microscope or stereoscope, and then we preserve this material for different purposes.
And one of the purposes is to not only of Lysodendorix sampling, but of all of the sampling that we are doing, is to build what we call a genetic sequence library. And that is because we want to make an overview of, we want to understand and make an overview of what species are actually living in this area. We can go to literature and we can find out what has been found before, so we can have an idea. As I said in the beginning of this dive, nobody has been exactly here before, ever, of humans. But even in the entire Norwegian Sea, there hasn't been that much sampling in the deep. The deep sea is very little studied because it's so far away from us. It's so difficult to work in the deep, and it's just the last 10-15 years we have been able to go down with our extended eyes, the ROVs, and look. So we have some samples, but not very many, from most deep sea areas that have been sampled at all. We don't have very many samples. And trying to find out which species are living here and trying to get a library of what their genetic build-up looks like will help us to later use less invasive techniques like environmental DNA.
So there's a rock, yes. A rock, you see that?
Yeah. But just taking a water sample or a sediment sample or an active filter sample and say what is the DNA here and how many different species, without being able to compare with a library of what those sequences connect to which species, we will only be able to say, oh, there is 25 or there is 58.
Yeah.
But we don't know what those 25 or 58 are, if they are species or if they are populations or if they are... Without that library, we don't know anything more than there are some differing genetic coding around here. So it's for the purposes of making these less invasive methods available to use, we do need to first build these libraries. So that is why you will see us sampling lots of different things to build up these libraries and make it possible to compare our environmental DNA with these lists of this genetic string belongs to that species type of libraries.
Yeah, indeed. Yeah.
And this is a thing we have been doing at very many museums around in the world the last 10-15 years now, building libraries of the nearby local fauna and flora.
Yeah, plenty of encrusting sponges.
As soon as we get some hard
Yes.
surface.
One fish there standing. Oh, again we have this beautiful hexactinelle. It's probably Roselidae, and then some encrusting sponges. So the different white or yellowish stuff that you see there, those are sponges.
And the one that has all the pink little dots in the end there, is that again the Lysodendorix?
That one here,
Yes.
yeah.
That
Just
was
trying to-
again the Lysodendorix.
And I think there were also some craniellas. Some of the
Brain sponges.
guys that always turn here and appear in these communities.
So this is again a steeper uphill. So we are starting on
I think we're going to be able to...
a mound. And this was one of the things that we wanted to look at.
Yeah.
So-
Let's just wait a little bit because of this area.
When we
with
planned
more,
this dive,
rocks. And this is
we looked at
perfect
these
for us to find the
little
specimen
hills in the seafloor and decided that they could be an interesting thing to
Okay, right, let me know.
dive over. So now we're starting to go up the hill of one of these small mounds or relative small mounds that we found.
Look at that.
At
When you
the
zoom in,
perimetry.
there's plenty of life. You can
Yeah,
see
look at that.
plenty of life at the sea bottom. Not,
Definitely, it looks like little stars hanging out there.
I think those are crinoids. You see?
You think they're crinoids? Yeah, that might be.
Stalk crinoids.
And maybe we have a crinoid field and not a worm field. I like crinoids
For
a
that,
lot.
Yeah. For that, we need to stop at some point and collect samples.
And I
Oh,
think
look
we
at
have
those crinoids.
crinoids here.
Yeah.
They are non-stalked crinoids hanging out on the rocks, and these are definitely volcanic rock.
Yeah.
You can see how the lava at some point in time has just poured out and gotten cold from meeting with the cold seawater.
Again, these sea lilies that you can see in the image. Top left. Now it's going to be
They're great.
passing.
Look at they.
Yeah, you see? There's plenty of them.
And these are animals that are sitting and picking up little organisms from the water. So all the time it seems to be a heavily sedimented area where we just see some of that volcanic rock sticking out every now and then. And I must remind you all that this is a quite steep uphill that we are doing, which would probably make that sediment a little bit unstable. It's always the possibility of rolling down. But we have loads of what we now think, if we could just have a close-up look, maybe, stalked crinoids.
Yeah, I think so.
Crinoids are part of the echinoderms, and they are close to sea stars. So if this crinoids, we would have a field of close related animals to sea stars standing on stalks in the sediments.
You see this one?
But it's quite hard to see. Maybe some of you who are watching can have the possibility of zooming back or stopping the video and looking at it again, which we don't have here at all. We're just sitting and watching it live, which I guess most of you are who are looking as well. I think, yeah. It is not super easy to stop if you have a very steep uphill. Then it's not easy to stop the ROV. So that is why we are continuing flying.
So there's this.
But there is a nice. It's almost like a little break in the sediment field there, right?
There's lots of organic matter and organisms being suspended in the water column.
Which is also showing what a very rich, energy-wise, very compared to other deep sea areas. This is
the spaghetti.
More spaghetti, so we have more
sponges,
of the Lisodendorix. I think that is going to be the sponge name everybody are going to learn within the dive
That's easy,
today.
Right?
And Lisodendorix complicata. And that is another ctenophore there, right? Floating up.
Yeah. Let me just check. Oh, look at that. This
shrimp
just appeared.
Swimming by us.
I think this is a fun-shaped sponge. The one now close to the lasers.
And a jellyfish, or
Yeah, I think so.
or a ctenophore.
I bet it's a jellyfish. I think we saw something similar passing by. is a lot of organisms flying away just in front of the camera, which makes things a little bit more difficult to identify.
Yeah, but we are going still slightly uphill.
So it's now 2,700.
And 35 meters.
Yeah.
And we still have a sedimentary seafloor in front of us. Still slowly climbing up.
I just try to.
The sandstorm.
So for us, it's really quite more appealing, I would say, to have rocks. Rocks are they for longer periods, not a sediment. And in rocks, you can find different species. There are sessile organisms that they got attached, and they produce. In some cases, this is one of the goals of this expedition, trying to find ecosystems that are like important for ecologically, producing being like small trees that form kind of forests. These forests, they boost biodiversity in some of these areas. It's not that this not flat, but this sedimentary area is not diverse, but
It
organisms
has a different diversity, and as we talked about, very much the smaller animals.
We have reached the red dots.
Yes.
Should we change the direction of transect, or do you want to continue?
How flat-ish is it here? It's not really flat, is it? Can we?
The slope is going to our right.
But it's still a little bit of uphill in front of us, yeah.
Oh, well, according to the Polyvene data, it's downhill.
Okay.
So it's now the very summit, or close to the very summit.
Yeah.
Yeah.
I'm not sure if the lines may be slightly off, maybe it was more to our port side.
Yeah. Maybe we can have a tiny little look here at the top, and then we can continue the line.
Yeah.
The top would be probably to our left.
Yeah. Is it possible to have a little look what the top looks like, and then we can?
Bridge RV.
So do you want to come to the left?
Can we stop for now?
Head up slowly, just get it.
So we're just going to have a little check about how
Okay, thanks.
how the top of this mound looks like, if it is more sand or mud, or if more of the rocks are peeking out at the very top.
And yeah, we're gonna have a look around.
And this is all we can do on this dive, because we are looking. We don't know anything about this area, right? We are the first humans ever to have a look here.
Yeah.
And you guys online are sharing that first time ever humans are looking here.
There's some rocks here. look. Yeah.
Look
indeed.
at those volcanic rocks and like lava flows that have come down there once upon a time. But with all that sediment on top, that is
Let's
some
just check.
time ago.
Who's there? Oh, yeah, this
Yeah, that's
fish
a
that we
nice
saw at the very
fish.
beginning, right?
Now we can zoom in. Look at that. Pink fish. Slowly swimming and finding food at the seafloor.
Quite big sponge.
And
to
there are
the-
some large sponges here as
In
well.
the middle of the image, you will see in a minute some details of this sponge.
And crinoid.
Crinoids. And this is
Oh, that's big.
something around half a meter or something like that.
Oh, wow. It's been there for some time, huh?
Yeah.
It's really been there for some time.
And again, the sedimentary habitat.
Yeah. And
seems to be decreasing on the sea.
and again, more of this sedimentary.
We certainly need to,
Yeah, they look
Identify.
like stalked crinoids.
yeah,
I think at the moment that is our best theory.
Because I can't see off here.
you see, you have a stalk and then they have like a,
Yeah, like in an umbrella sticking
umbrella,
out.
yeah. with different, it's not a, it's covered, but they have like a different tentacles. Well, I wouldn't say this is tentacle, but for you to understand. And they're capturing food from the water column. Oh, look at that.
That-
It's a ctenophore.
That's a ctenophore. Yeah, that's a pretty ctenophore. And more lava rocks again. This is a strange hill. So several thousand years ago, this must have been just rock and probably quite new. I don't know. Now, you geologists out there, please don't beat us up for this. We are biologists and not geologists, but I think this looked like a place that was very active several thousand years ago.
Okay, can we keep going with the movement direction 045 at 0.3 knots?
How about we try to sample?
Yeah, if we find a flat enough place to land.
Yeah. So we try to sample?
If we find a place that is possible to land. But
Just let me know if you feel it's too difficult or whatever.
yeah.
Now that we have rocks, we could potentially stay in a place where this.
What, do you want to go close to some rocks like this?
Yeah, but first we need to identify potential samples that are
Or you just tell me.
in our interest. Yeah.
I have to again say we have the most lovely pilots who are doing the undoable for us many times every day. So a big shout out to our lovely and very, very, very good pilots who are helping us doing this. We're just sitting here looking at video footage together with you guys, and they are doing a lot of work to fly this machine for all of us. So
Yeah, indeed.
thank you guys.
Yeah, very well.
And we always want things, and they say, oh, maybe we can make it happen. So it's lovely.
They make it happen. Yeah.
They do make it happen,
They are really good.
if it is at all possible.
So 2025, the right spot now for our first.
Sit down and check. Yeah, but it's then we have to find a good place to sit down.
Yeah. Okay, some rocks ahead, but just small stuff there.
Maybe we are getting toward the top of this thing.
Look at this.
So we can use another beautiful. That is a jellyfish. Yeah.
Bye-bye.
So we're using the sonar of the ROV to see where the landscape is going up and where it's going down in front of us. And it still looks like it's possible a little bit more going up. I should say we are now at 2,708 meters, so we have been climbing quite some meters since we thought we were at the top. And of course, even though we have really strong lights on the ROV, they don't reach very far because they have to go through all that water. So mapping visually at the seafloor of this depth is like mapping your own country using a handheld torch in the middle of the night. You only see a very, very short distance ahead of you, and this is what we are doing now.
Oh, yeah. We can just wait until we get to an area with many rocks.
Yeah, and this is
Less
where
than
the
20
sonar is getting a little bit more help for us than the lights, because the sonar tells us there are some rocks coming.
I can see it now. So I'm gonna try to identify specimens, species that we are interested in.
Yeah, look at that. Oh yeah, a rock wall all of a sudden. Out of this sediment comes a wall of rock. And look at that, must have been such an immense lava flow once upon a time. Wow. Look at that, that's cool.
Can you watch this map? So probably now we are just on this hedge.
Yeah.
This is another wall, another type of terrain. I don't know if that's your intention to go through here or.
Well, we want to after a while to go towards, because there's another mound like this.
Yeah. Because towards the line, you mean? Because I think we have passed this mount and we are now going up this hill. So if the intention is
then we
hill,
can see if we can do some if we find a place where it's possible to either land and sample or hang and sample. This you have to tell us if it's possible.
Sediment or
Most rocks.
Close to some rocks, yeah.
Now that we've been through this area with rocks, we didn't identify any of the sponges that we are, or creatures that we are interested in.
Yeah, if you see any rock with what you want, just give us a shout.
Problem is that it's like images are like too dense in here in order to identify in advance what we see. What's there?
That looks like one of the lollipop sponges, right? No. Or is it? Does it move faster? No, it's
this is our beloved Lysidendorix, is it?
sitting on a small rock.
Yeah.
It's a small rock with
Can we stop here?
Lysidendorix.
Because this way we can just pick
Yes.
this, and this.
Yeah, I need to see where I am, please.
Sorry. That thing on the on the left.
So we will make a little bit of a sandstorm when we are landing, and very quickly, maybe.
Do you want to pick it up with a minute?
Yes, I
need
think.
to know so I know
Yes.
where to.
Yes, I think.
Maybe the slurp, do you think?
No, not the slurp. I think. Yeah. So we're trying to land. Our pilots are trying to land so that we can pick up this rock with the Lysodendroryx, the species name of the day. So it's almost like we're going to give you all guys a test at the end of the of this live sending, and say which was the species you had to learn today. So it's all Lysodendroryx, but we are going to try to sample one because, as Sergei talked about, we're going to find out how genetically connected the different populations of this species is, because they live everywhere.
Yeah.
It's in this water, so in the Norwegian Sea they live everywhere. And to understand how a species that cannot travel very long distances, because I mean when they are adults they are sitting someplace and not moving around, they are sessile as we call them.
Yep.
And when they are breeding their babies in addition, how can this very widely spread species be genetically connected with other specimens of the same species? If the populations are genetically distant, or if they are very closely related, then we can start thinking about how the contact is actually going. So this is the question we are trying to answer with this sampling. And then, since we are now landed here and waiting for the sandstorm to disappear, we will hopefully also be able to take a good look at those things that we think are stalked crinoids.
Indeed.
And maybe have a little bit look around about at some of those. There were small rocks, and it's amazing how just this tiny rock in this sea of mud can be the home of a completely different habitat all of a sudden.
Yeah.
So this is showing everybody the difference between the hard bottom and the soft bottom animals.
Now we can start seeing our target.
Maybe.
To the very bottom.
Yeah. So we have to do a little bit of positioning as well, and then we make more sandstorm around us. But it will land. It will all be better.
I mean, the strong currents that we're dealing with are gonna make the job of cleaning this plume. It takes a little bit, but it will be like super clear in a few seconds, so that you can see how pilots are gonna be surveying these organisms that we are interested in, and putting them into a bio box that you will see, so that you can get an idea of how is sampling in a depth of nearly 3,000 meters.
2,705 meters to be exact.
Not too
right
bad.
now.
Sorry? Yes.
So this is going to go away.
Yeah, so
This is going to go away. This sound.
Yeah.
Yeah.
Directly down.
Absolutely.
So there's your
So-
rock.
Do you think we could potentially take the rock?
Yes, this is what I'm thinking.
Yeah.
Where are the lasers? How? Oh, yeah. So it is. Yes.
Can we bring the sexton in to do these operations?
Yes. Yes,
I
let's.
think it would be a good
I
idea.
think we need to bring in the sexton to the other camera that we are seeing here because it's going to be in the way of our sampling. So we will take it in.
Thank you, guys. So we'll take a close up once it's a little bit more clear.
Yeah.
Yeah.
We'll take a
The sandstorm is
some time to
landing,
identify.
or maybe not. Yeah, it's landing. It's
It
sort
will.
of
Some-
slowing down. Yeah.
Okay. Take a few more seconds for the water to clear. So guys, I believe we can take the rock, the whole rock, unless it's not connected to a pillar.
Connected to the planet.
If it is connected to the seafloor, we don't take the whole rock. But if it is just,
Where would you like it to go? Which position?
what
I mean,
is the
closest to
easiest for you?
closest to you.
Or we just hold the port door? Where do you prefer to reference?
Can be the port.
Yeah, so to start it. Right.
What do you think we're going to be able to? Is better
Port
with
front,
the
yeah.
scoop or?
Port front.
Port front, please.
Right.
Left front. Oh, right.
Oh,
Port
yeah.
front, right.
Okay. Yeah.
The one on the rock, you mean the whole rock?
The whole rock.
Yeah, because it's not that big,
Yeah, we can just
Port
grab it.
front, right.
right?
Yeah.
So now
maybe
you
like
have
a
to, yeah,
10.
10, 15, 20 centimeters. If the rock is what we see,
Yeah.
if it extends down to the center of the earth, we are not bringing it with us.
Well, give it a try and then
We'll start.
We give it a try.
So we put in a port drawer. We've got one of the coffee mugs in one of the compartments
Uh-huh.
at the front. So whichever one's not populated, we
Yeah.
really empty that one.
We will put it in port drawer. I will also try to write it here. I have taken the notes.
Yeah. Yeah, a few more seconds, hopefully, to get a clear view on the species that we are going to be sampling now. So as a reminder, I mean, we've been talking about this. I mean, taking samples it's super important from the scientific point of view, so that to identify different species, connectivity and stuff like that. And given the how, I mean, given that these organisms that we're going to be surveying in here, like relatively abundant, they are quite abundant, I would say. The effect on the population, it's going to be negligible. But of course, we need the specimens for these studies.
And we make sure to do as much as possible with each specimen that we are sampling. So it will not just be used for one thing, but it will be used for many things. And another of those things, everything we sample are going, or maybe not every single Lysodendroix, but yes, also that will be going into a library, a genetic library of the species, so that we can build a library for being able to identify the species later as well, from genetics.
It's taking a while to clear.
Yeah, that's how it is when it's very, very muddy and very much sediments all over around us. That is what sampling looks like in the deep sea. You have to have patience when you are in the deep sea. Not only does it take a long time to get down and go up again, but also to work. It does take a lot of time. And just to remind you, not only are we very deep, we are at 2,700 meters, but we are also at a quite cold place. It's minus 0.68 degrees down here.
say it's a little bit cold, right?
Yeah, I'm not sure if it's my bathing temperature. Do you have a limit where you start bathing? You come from Spain, so you need warm water, huh?
From 20 something.
From
upwards.
20 something and upwards. Yeah.
I'm not used to cold waters.
Norwegian children say as long as it's 2 digits, it's okay.
Okay.
So from 10 and up.
You're so brave.
We're just stupid, probably. Exactly about that one. I don't know. We have a wide variety of nationalities in the shack with us, so we can check. What do you guys think of at a good temperature to go swimming? Does it need to be more than 10 degrees?
If you live in the UK, no. It's too cold. Yes, no, it would be nice.
It would be nice if it's a little bit more than, so you would also not go swimming at minus 0.5?
No.
No. Okay, there is an agreement in the shack that minus 0.68 degrees is a little bit too cold for our swimming.
I think we'll probably die.
Nah, but you would jump out again really fast.
You can see it with any of the camera from here.
This is extremely fine sediment, very muddy, very silty probably almost. So the only camera that is seeing anything at the moment is the one looking straight down.
Yeah. It's super clear now.
Yeah, because that's nice and calm. But every now and then we can see the front of the RV. So we are hoping that this sandstorm will land soon. It also is talking a little bit about how much currents there are around here. And maybe a little bit how much extra current we are making.
Give us something to identify. Still to get it clear. I can start seeing something.
Maybe not. Do you have an idea of how long you want to hang out? How precious is this rock for you?
Well, given that we've been a while, I would definitely wait a little bit longer.
Yeah.
So the idea is to pick the rock, but also these crinoids.
Yes, we would like to have these
stalked
stalked crinoids, and we definitely think they are stalked crinoids. One very, I would say, interesting thing about these stalked crinoids is that for a very long time we knew them only from fossils. So people thought 150, 200 years ago that these animals were prehistoric. But they are living and thriving here. I mean, have you seen that field? It's crazy, right? It's a stalked crinoid heaven, if this is what we are seeing.
Yeah.
We have to take that because we have still not been able to properly zoom in on them. But they are definitely thriving in the deep sea. So they are definitely not only prehistoric, but there are some beautiful fossils of really large stalked crinoids around there.
Yeah.
Some really famous big fossils of things that are almost as tall as we are.
Yeah. So many millions of years ago, those stalked crinoids probably were dominating the
Rulers of the sea.
sea.
Yeah.
But now you can mostly find them in deeper waters.
On soft
It's one of the
seafloor.
examples.
Yeah, on very soft, muddy seafloor.
How quick you could do this? Sorry? How quick you could I mean, I can't see us getting much better. When we do get
No.
fish, maybe just try and pick it up pretty quick.
Yeah, we can.
But now it's clearing.
Yeah, I think we're gonna with the.
It's actually
soft
just.
seabed, I think.
I think this is as good as it gets.
Now we are starting to see.
No, I think it's going to be maybe see and not see again. Sorry, but I am a little bit pessimistic about this sandstorm.
We'll actually move outdoors options.
I would put the T4 cam on your
But our amazing pilots are suggesting to try to sample anyhow. This is something I wouldn't dare, but I am so pleased we have the amazing pilots with us today.
yeah.
And we're going to do a grab more or less in the blind, and then we can continue after that.
Two. Yeah. Is this one here?
Yeah, that one. Oh wow.
When he picks that up, this camera, I'm going to put the drawer out.
Yeah.
Can you just zoom in so you can see where to
Yeah,
put it, please?
sure.
'Cause
Yes.
I've got nothing on this camera here, please.
Our pilots also don't see very much, so this is going to be
Yeah, we're just seeing something that you are not. It's the arm trying to get the rock with the other specimens.
so we are sampling more or less in the blind here.
No, it's not. It's not. It's attached to the planet.
The rock is attached to the planet. Oh, we've been waiting this long, and then the rock is attached to the planet. Ah,
Well, there
Could
we
be
go.
the drain plug as well for water.
oh yeah. You think if we pull it, the water will start pouring out?
Yeah, I think so.
Yeah.
Nice.
Rock piece. I think you are not interested in the rock itself.
Not really.
No. Ooh, that was a bummer.
Yeah, it's bigger than we thought, right?
Yeah.
Never mind.
If it is bigger than we can pick up easily, and we don't see a thing anyway, I think. Ooh,
There is a piece that I broke. If there is any
Yeah.
life attached.
so we have a piece of rock in one of the manipulator's hands, and we're going to bring that to our bio boxes in the
You're gonna see.
Do you want that piece?
No, it wasn't the one with the sponges, but yeah, you can just.
yeah, let's put it in the bio box, and then we can move on.
Yeah.
Yeah. Oh, we
Well,
see
that's-
the hand coming out of the shade here.
So I've put the drawer out and zoom in so maybe you can see it.
And we zoom in also,
Okay. And then the drawer.
so that we can see
Okay, extended ports tray.
the port tray. And zoom in on that.
Okay. There you go. And that's it. Front right there.
You see?
Bio box 1.
What's it? 2.
Okay, so it's bio box 2. We also don't see anything here.
also the arm that our pilots are using to grab samples has done its job.
Yeah.
Shame it's too. Yeah. Thank you so much. It is somewhere else because it's too much
Yeah.
sedimentary pollution here.
Too soft.
So I say we try to move on to follow that next transect. So we call this the end of this first transect, and then we move on slowly. We don't need to go back to the line and then follow it, but just sort of merging into the line, the next line, so that we go a little bit down towards the line.
So apologies for everyone who was waiting for the sampling to be done. Well, it was done partially. We took a piece of rock, this attached to the very bottom, and then we'll bring it up and we will study organisms and also trying to identify the characteristics of this rock. But for now, we're going to be continuing with the transect.
So, would you like me to head back down to the line?
Yeah,
Or
just
do you want me to
sort of,
continue along here?
if you sort of lean into the line after a while, you don't need to go straight down and then
Straight
have
down.
a
But so at an angle
yes,
come down.
at an
yes.
angle come down to the line. So we
Okay.
just call this the next transect.
There is something like
hemos subido ya el primer montículo. ¿Cómo estaba? Terreno arriba del primer más rocoso.
okay. So maybe we
And now there is a lot of things happening at the same time because the next shift is coming to take over in 6 minutes.
because no one can stay on
And
this side
we
of the-
are ending the first transect and starting the next transect. So there is a lot of logging to do and a lot of chaos behind us.
The ship is one
But we are
Oh, yeah.
on our way to hopefully see something nice and interesting along the next line.
very slow. It's likely because
So it's ship's side. Just
And as I said, we are at 2,705 meters depth.
okay, I do when with the ship in 1:30 to come to this
right,
side.
yeah. You do 120, you do 130. Yeah.
So we are going to do an almost
Or I can move 090 for now until we
almost
get the right position and then I
straight
start going
eastward, a little bit south
to let you
again,
on the safe side
Yeah.
as soon
but
as
mainly eastward
Yeah, correct. So I'll start
transect
over at
now
130.
to
And I can
another of these
090
hills
as soon as we're
to see if it is
Yeah.
as muddy or if it is more more
ROV.
if there is more volcanic rocks there, if there are any other
Okay, we are
beautiful
ready for the
sponges
next
to see.
movement. We would
So,
go to
with this
direction
next transect, we will
090
also
at 0.2 knots.
let you have the next shift to take you through that next transect. And that will be Julio and Franzi. And if I know Julio, he is going to say we need to sample. So as soon as they find a place to sit down again, you can fight the sandstorm and see if you can find a place where you don't want to sample the entire Earth.
As we eat.
Because Sergi, he wants the world. But it's a good thing to want the world, right?
Yeah.
You just, we can't
Just a
always
small
have
piece, not the
everything.
So when we're flying away from this area that was packed with this sedimentary plume,
Can be useful.
just because we have, I mean, this is really fine sediment. I think we'll find another spot.
But we bought a couple of years ago.
So we will turn the RV around and fly towards our next transect. And if we look at the map, I think, did you have it? Did I have it?
I think it's there, right? With the
There,
transects.
yeah, it's approximately,
It's like 300 meters or so.
it's a 300 meter until the next mount. So we can also think a little bit about what happened back in the day when these areas were active volcanic areas, because all of those rocks that we see are definitely old volcanic outspewings, I would say. So lava that has become rock when it met this very cold minus 0.67 at the moment. We can ask the new shift, would you go swim in that?
For sure, somebody swimming? No.
Yes.
Julio is coming from Mallorca, so it's not going to be his common temperature of swimming.
With the submarine.
Oh yeah, with a submarine? Well, we are now with an ROV, and I'm very happy to do this with an ROV. It's 2 minutes until cassette change.
Can be confused. The altimeter can be confused by the sediment.
How about you, Anne, then? Do you go ice swimming as a
I-
pure Norwegian?
I have done it a couple of times from the Arctic floating ice, so the sea ice. It is not a long swim,
No.
but in Norway we have this idea about if you can do 3 swim strokes, then you have been swimming. So there was sometimes when we would have, we would anchor the vessel into the ice, sea ice, and then we would all go out on the sea ice. And one of the deckhands would have a rope around one of us, and we would jump into the water and get helped out again. It was very, very
so
cold.
brave.
It's not brave as much as stupid, I think.
Yeah. Didn't want to say that. So
I can say
ours-
it about myself, right?
So it's
T2. Oh yeah.
The rest is fine.
But I also have to say, I think I've stopped doing that. Last year, I only tried 4 degrees at Svalbard.
That's more.
And that is a little bit sad, actually, because this is, of course, to do with climate change, that the water temperature is rising so much there. So, making it possible to go swim.
Oh, yeah.
Flying away from this area.
Yeah, we are flying away from the snowstorm. No, that's not the snowstorm, the sandstorm.
Can we go close to the seabed on this? Or do you just head down to the line?
Oh, no, yeah, we can go.
Is it possible to go close to the seabed?
I guess it depends on how.
Yeah,
Yeah, I was going to be up and down all the time.
Yeah.
yeah, that would
Yeah,
be
otherwise
nice.
we're losing
Yeah,
the view of the organisms in there.
So do you want to go low?
Yeah,
yeah.
as close as you can.
So 10 seconds to.
Okay, start recording again.
Yeah. And that means Sergi and Anna Helene are handing over to Julio and Franzi.
Yeah.
And you guys out there enjoy, because now you're getting the real team coming. Well, one real team is leaving and one other real team is going. So let me just log this.
So more exciting views coming up. Once this dust is nice left away. Now, yeah. So,
Okay.
thank you so much.
And yeah, so crowded here, we have to get one out of the chairs at a time.
Hello. So Julio and Franzi are on shift now, and also starting a new transect, hopefully. So yeah, ready to go further, exploring more of this volcanic area here.
In parallel to
Right. So it just keep
And seeing what else we find.
Okay.
Well, fine, like that was Los Grass and
Okay.
maybe interesting. Yeah, wait.
So yeah, on this trajectory, you happy
We're
with this?
currently discussing with the rough pilots also, because we're on an exploratory dive, we can experiment a little bit also what we see in our bathymetry data, so the topography of the seabed, what we think is interesting to go along, and we see, for example, some cracks, some crevices, some valleys in this area that look interesting on the map at least. And yeah, since we're the first ones out here, we just want to have a look there and see what it looks like on camera, and not on this bathymetric map. Is it good?
Yeah, I need a pencil.
Yes,
I have.
We have multiple pencils here by now.
Okay.
All our paperwork.
So lots of tools there, yeah. All good. All good.
Proper office here, I'd say.
090. Now you are
So,
will clear your tracks now.
Okay.
What's the direction that we are going in? That I think is good.
do we have
This is very
some
sediment-
holes in the sediment there? Or is
Very
it?
sedimentary, I'd say.
Actually, for me now we are going more like south. 0.7.
Yeah.
That's a hole.
Some
There's
holes
somebody living out there.
Yeah, even though we mainly see sediment here, it doesn't mean that there's no life here. A lot of life actually lives in the sediment or below,
But the thing is, we were relying
or
on
small
science.
organisms on the sediment. But we cannot see everything with our camera here. But we're trying and zooming in really close on what could be. Could it be a sponge?
That looks to me like a big sponge.
Woohoo.
Bridge ROV.
Let's see if I can focus this.
Can we slow down to 0.1?
Yeah.
Just for us to understand.
Maybe we just have to get a little bit closer.
Yeah, that's an Asconema.
I'm logging that already.
Asconema mega atriada.
Ah, yes.
And then we have the community of little sponges growing on the rock. This is where potentially the new species is.
But it's too big to sample.
That's too big, so we are looking for a rock that is loose and a little bit smaller in size, so we can grab it with our hand.
So on one of the previous dives, we sampled a little rock and we found potentially undescribed species on there, right?
Maybe, yeah, looks like. looks like we still need to get DNA out of it and to do a lot of work, but it looks quite promising. It's a genus that is not known so deep, so that makes things a little bit easier in order to tell that it's new. That's why I feel that really is a new species, because the deepest that we are aware of was found around 700 meters, and that one we found at 2,800 meters. And it's also the first species of the genus reported from the Arctic waters. So it looks good, but we'll see. One of the goals in this dive is to find that sponge again and to see if we can get specimens in formalin, so we can see the cells.
Okay, can we change the direction of this movement to 150?
So that's why we're on the lookout for some little rocks where we can hopefully find this and shed some new lights on the species diversity down here.
But as soon as you go to those deeps, it's quite common to find new things. You have to think how difficult it is to sample here. You need to come so far with a big vessel. So historically, this kind of depths were not available for people to sample. But now we have this technology and we can start to understand what's going on.
Which speed are we going at the moment? Which speed?
Point 2.
Okay.
That's point 2.
Maybe even for these parts from our side at least we could speed up a little bit, or rather
Well,
not?
now the rocky outcrop is coming, maybe. You see it there?
yes, true. We also have a echo sounder. No,
Sonar.
is that sonar?
Similar.
I always get confused. A sonar here to see like what type of bottom is coming up, and we see some rocky area coming soon, and we can also see it on the camera. So we'll look at that.
So we are
And a little
1.5.
jelly going through the picture on the left.
Yeah, it's fine for the moment. Later if you can go a little bit closer, that's fine, but it's only when you
Yes,
forward,
yeah.
so.
Do we have little lollipops, or are those the... No.
I'm not sure. That would be hard to say.
That's
That
more
looks
like a worm, no? Or like the crinoid.
hard to say.
Yeah.
Hard to say. We could
Something's living there. That we know.
Potentially try to collect one of those, but the sediment is so muddy that it's a nightmare. Every time you land, you get this cloud, and then it's forever. So I think that
we want to be really mindful about our...
We should try to find an area that is flat enough so they can land, but it's rocky, so we can have some visibility of what's going on.
That sounds like a good plan.
So that looks to me like Hemigilius, Arcopherus.
Is it in our list? So we can just
No,
click on
I
it?
don't think so, because we identified this species 2 days ago, so it's not,
Oh, it's a
not
very
in
new
the
one.
list.
Amigellus? Copherus?
Arcopherus.
Arcopherus.
So you put an H at the beginning?
The silent age of the Spaniards. again.
No, here.
Yeah. Hamigellus.
Yeah, perfect. Arcopherus. It's fine.
You will know later on,
Yeah,
hopefully.
no worries.
Do we have more? Oh, no, it's almost gone. Thought I saw more interesting sponges on the lava.
Oh, yeah. That's what we want. Rocks. So Francie, we will try to clean a little bit.
Mm-hmm. Yeah. The area is coming up that we saw at the beginning, just a few minutes ago, on our map, with some cracks, rifts on the surface, and we're looking at these rocks now, or starting to look at them, climbing up a little bit.
You see all these little dots that grow on the rock? So probably our new species is
one of those.
One of those. Yeah, we'll need to get to be much closer.
And which sponge would that be?
That's an hexakinelate sarcom. I don't remember now. It will come up. I think it's sarcocalids. But I might be wrong there.
Because Julio, here's our sponge expert, or one of our sponge experts, next to Sergi, who was just here, and Paco, who will come up later likely. And a little Ctenophora, a little jelly.
Yeah, you see that it has a stem?
Mm-hmm.
Try to catch it with...
Yeah, one of our... Could you also call it lollipop because it has a stem, or is that just for the?
Yeah, I guess.
It's not
I mean,
so
it's really big.
Yeah.
So it's a very large lollipop.
Mm. Wow. Is it known why they build stems? Is it to be far away from the sediment and then have an easier way of filtering?
Sure.
Can we change the direction of this movement to 120?
So, hold on, wait.
Yeah. Let's coordinate. We have crinoids there now. Yeah.
No, I mean,
Yeah, many of them hanging out together.
I can only go off that.
I was building a ladder or so.
Did they saw crinoids in the
Okay, thanks.
Sergi and Namen?
Sorry?
Did they saw crinoids? Sergi and Namen?
Not sure.
So yeah, stems are to stay away from the mud. So you don't want the mud because the mud will block your aquifer system. So you build a stem, you can attach to the bottom. That's very useful. And then the main body is away from the ground. So you can still filter feed.
Especially in those habitats here and these bottom characteristics.
Exactly. Well, you can tell when you see a rock, there are no stems on the rock. Everything is
not needed
like
there.
a crust.
Yeah. Could you zoom in on some of the
Sure.
of our bottom dwellers? Yeah. Hard to focus there.
Yeah. So with movement.
Ah, there are little critters sitting on the stem.
Yeah, there's a couple of things here. Our colleagues were mentioning that this may be a crinoid,
Yeah.
but in
so
this
a feather
case,
star?
there's something growing on it as well.
Mm-hmm. Maybe some of Annalina's beloved amphipods.
Maybe, and I think I saw a brittle star over there. No, it
Further
went on.
to the right?
Yeah.
That one?
No, there was a really tiny brittle
Oh,
star
okay.
escaping, but couldn't see it.
So we're still climbing, and we see some more rocks coming up here. We're at the moment in a really deep area of Mons Ridge on the mid-Arctic ocean ridge that spans all across, like is part. Oh, hello, hello, jelly helmet.
Right.
Yeah, we're so at the moment on the mid-Arctic ocean ridge, on the Mons Ridge, part of the north of the Atlantic ridge that spans all the way across, and it's a very, very slow spreading ridge in comparison to some other ridges that we have, so for example in the Pacific. And it means that the tectonic plates here are drifting apart slowly, and there's volcanic activity underneath, and this is also where we... Yes, an enemy. Oh. And
Here
some
again,
more
the
sponges on the
community
rocks.
of- yeah.
Yeah, and here we see the remnants of the volcanic activity with all the lava rocks.
The orange one, I never seen before.
Wait, the
That's-
orange one?
Yeah. Okay, we see something similar, we try to stop.
Maybe on the one that's coming up, there's a lot on it. But
The thing is that we need a small rock.
yeah,
It's gonna be tricky.
But there's Lysodendrix on there, no?
That's
Just
Lysodendrix.
for logging.
What do you think about stopping around this area? Would it be easier or still rocky?
You're certain of the sea there. Yeah, we will positive.
Maybe better on the rocky than on the muddy, or that's the problem?
We'll probably go on the rocks, yeah, I'll go and see them.
Then it's fine.
So where would
So
you,
because we
where would
wanna,
you like a sample? Tell me
sample
first,
more
please.
of the Lysodendrix and also have
Let's keep going.
a small rock with what we believe are unknown sponge species, undescribed sponge species. We're looking for a good place, a good spot for stopping and for taking a small piece.
See that rock would be good, but I think we can.
Yeah.
I prefer
But
to
is it
wait a
detached?
little bit. Yeah, that looks good, but it's tricky for them.
Yeah,
So
it's very not easy to stop here with our big ROV. These are also interesting crusts on those rocks.
it's possible. Just asking. So if it's possible there.
Yeah, we can see again how easily the really fine sediment is
See all the thrusters.
swirled up here.
Yeah, that's why I was.
We had a problem last.
Yeah,
I can land there.
because
it's
it's
not bad.
like a fine powder,
We wait.
So it goes to the left, right? So if it goes to the left,
but it's
I
only
should try to spot something
very
that is
temporary here.
on the right. I don't know if that makes sense.
Yeah, I don't have a problem.
More rocks piling up here.
So you rocks are in this one.
The last time we didn't work very well. So we can try again.
So it has to be mud. It cannot be, right?
Julio and the pilots are discussing a little bit what could be a good area.
We got the box we need to push out.
Yeah.
Keep everything exposed.
So us two over here, we're mainly logging what we see, where we can steer the camera that you can also see with our
Where would you like,
joysticks.
what you want to see?
But
What you
the
want me to stop?
real work
Well,
of
actually,
the steering
a little bit of this
and the handling of the
because it's moving
of
now,
our
I'm not
robot
sure,
are
but
the people over
Yeah,
there
we're
doing.
looking for a rock that is not big, so we
yeah.
can collect.
So they're
For instance,
the
here we have
real
some.
rough experts here and guiding us through this unexplored landscape.
Where do you want me to fully
To collect rocks today, is it
Yeah.
for?
So we're settling down now?
We're trying, but we have
No,
problem
we're
with the
not
map.
sitting down yet. You want me to sit
Ah.
down here?
Okay.
You're not going to see anything now?
So yeah, we're not even settling down, but
Well, you're waiting a long
nevertheless
time.
the sediment is so fine that it's just stirred up easily.
I can stick a stump, but I'm just.
If you find a rocky place that you say, I can land here, then you tell me. If not, then
We can, if we see some fairly level rocks, we'll try and down there. We've got to be careful.
So we'll keep going because that mud will take forever to settle.
Relatively, yeah.
The problem is that we cannot land on the rocks because the ROV has cables and has a lot of devices
Yeah.
that could potentially damage. But our pilot will try to find a place that is maybe a little bit less muddy and good for them to settle.
That sounds like a good plan. What do we have there? A bigger sponge in the distance?
Yeah,
Ooh. Interesting shape.
that's one of those Hemigilius, Arcoferus.
Ah.
They are really common. And these crabs that are a little bit larger, that's Imidesmidae. It's a family of demosponges.
The demosponges.
Imidesmidae. Will be a
As I
little bit-
learned, the sponges of the people, because the sponges that are sometimes used also in the bathroom or in the kitchen can be made of demo sponges.
You see that? That,
Yeah,
that looks
the
like
one in the middle.
Yeah, that looks like Forbas to me.
Like what?
Forbas.
Forbas.
P-H-O-R-B.
Do you want us to stop here?
Yeah,
quickly?
perfect. Well, thank you.
Yeah.
Thank you very much.
We are coming to a halt.
Okay, we've just stopped here.
Ooh, and we see nice rocks everywhere.
Bridge, ROV.
Go ahead.
That's perfect.
Can we stop for now the movement?
Okay, now we take our time. There's some sediment. We will wait until it settles. We will have a nice view of everything. We'll think about what we want, what we don't want. I saw some little small rocks, very promising. So yeah, let's wait now till we got some visibility back. Rocks.
And lots of them.
Rocky Town.
Remember the program, Flag and Rock? That was always to be on Saturday.
And we have so far taken already one rock sample, right? That was Anne-Helene and Sergi took it.
Sure. Was that it in the end?
Oh, I'm fiddling with the microphone, I just realized. Oops, sorry for the noise that I potentially caused. I think they put it in the front drawer. Yeah. So we have one in there already. We have nice sheets here prepared for the different compartments of bio boxes, as we call them. So the different boxes for biological samples, and to keep track of what is in which chamber, where. We have printed out some templates and created us some nice paperwork here, which will be very important afterwards for knowing also when the ROV comes back on deck, because then also the real work starts for the scientists to get all the samples out of the bio boxes, out of the compartment, get them to the lab, get them with the labels that are correct, so it all matches up in the end. Do you need something?
Yeah, it's okay. I'm just
Okay.
taking note of the coordinates. Sorry.
That's also right in front of me is a screen with our coordinates and the depth. So we're at the moment at 72 degrees north and at 2,700 meters depth. And all is noted down. And we just need to be nitty-gritty about our logging here, also for the later analyses that will take place, and all of that.
It starts to clean up a little bit.
Sorry?
It starts to get better, the visibility. Not here. Not here.
I can move my camera if you want to.
Now we get a clearer view
Let me see what's
of some of the
going
rocks.
So maybe we should tell Jason that there's something interesting here when this clean up. Something about the new species.
Yes.
Maybe. There's still some time until this cleans up.
Yes.
What was that?
Yeah.
What was that?
Was that just a piece? I don't know.
It was like a crustacean.
Yeah, it could have been. It reminded me of a seahorse, but I was like,
me too. It's like,
what are you doing here?
what's going on?
While we're waiting for the dust to settle more, since I was talking about the bio boxes, we have different setups possible here with the ROF. We have 2 bio boxes in the front that can be divided into several compartments, and we have at the moment 4 on the left side and 4 on the right side, and we also have more compartments in the back, 5 more. That can be used as well. And we have therefore quite a bunch of possibilities to take different samples and be also clear in dividing them from each other, because sometimes we want to be very clear which sample came from where. Sometimes it's also possible to do that by, we have a shrimp swimming through the picture, by the way. If we have 2 very distinct species or samples, then they can also sometimes go in the same bio box, because then later on we will know anyway, that sponge came from that area and that other sample came from that spot, and we cannot mix. We really see nice here the eyes of the shrimp as well, the very ghostly eyes almost, yellowish white. I should look at this, by the way, and not just stare at it.
There's another one over there. There's another one over there. You see?
There. Yeah. There we have another pinkish dot.
Yeah.
Is it is it a Craniella sponge, right of it?
Where?
It's not visible anymore, but there was like this round little ball, but it could have been also a sedimented, I think it was more likely a sedimented rock.
Okay.
I start seeing sponges everywhere.
Yeah.
So
I don't think we have Craniella here.
no.
We
yeah.
may.
Yeah.
It's too deep.
Could be a surprise.
It's too deep.
Until which, so the Craniella is more in shallower waters, Not really shallow, but
not shallow, but
yeah.
But this is super deep. There may be canyelas that deep. I'm not sure about that, but not in this area that I know so far. So there are, in this community, there are Polymastia, there is an Aliclona, there is the Emigilius, there is the Stellodorids. I saw a couple of Imedesmias.
all on that rock.
On the rock that we collected the other day, and the communities is the same as we are seeing now. I think that the other day was a little bit richer, maybe, or more dense. But this may have to do with the sediment. So this station is more sedimentary.
And does that block the sponges from even settling there?
Well, a sponge wants food, which usually comes with the current, and substrate. So rock is better than sediment, than mud, for instance. That doesn't mean that mud is not okay, but then you need special adaptations.
Like a stem?
Like a stem, yeah. Some produce a lot of mucus, so when there is a lot of sedimentation, they just release mucus, and then the sediment doesn't go in. But ideally, you want rock and you want a lot of current bringing food. And because sponges are very well adapted to this kind of depths, they don't have so many competitors for space. Competition for space is something that is very widespread in marine habitats. So some sponges are toxic. That means that when they grow, they release secondary metabolite, toxic molecules that may kill whoever is growing next to you. And this is how they compete for space.
Cheeky little sponges.
But this mud is going on and on and on. It's getting worse. So we wait 5 minutes, and if nothing happens, then we move on. What do you think? We wait 5 minutes, and if it doesn't clear, it's clear.
I mean, if you spot something, and we can probably get it fairly quickly. Well, we've got to
before everything happens.
Yeah.
The thing is that I really want to close up.
Close up. Oh, right.
Okay.
Okay. Yeah. That's,
So for some samples we want to take, it's also important that we see before what we take, especially for the species which we don't know yet. Because then we can tell from the pictures or from the video how they looked like while they were still down here in the water, and compare it to the samples we have taken them, and have a, so to say, in situ comparison. And and that's important for the species that we don't know yet, right? You can probably explain it a little bit better, why we want to have also a picture of, or a video of the...
Because sponges, they change a lot the shape when we have them on deck. So what we want is to characterize the community. For that we need to match what we collect with what we see. So we don't need to be collecting all the time. At some point we will be sure that what we see is what we collect, but for that you need to take many close-ups and then to sample, and then you are like, okay, this thing that now I have in my hand is that little thing that I saw on the video. Like that you can then process the video, the whole video, without being collecting all the time. And you can be saying, here you have these species, here you have two, here you have more, this area is more diverse. This species that I found here is repeating in the net transit, these kind of things. So it's quite important. But I don't think it's going to happen now, because this is getting worse and worse. So maybe we will just move on and see if we can find a place with less sediment, or maybe where the currents are a little bit more nice for us to clean this cloud. The thing is that the ROV needs to have the motor on constantly, and I think that the cloud is happening and happening and happening because we have the motor on. So
Yeah, so we have these propellers.
yeah, exactly. Propeller. And on the other side, we cannot land on the rocks because it's dangerous for the ROV. Many cables hanging on.
Yeah. We don't want to get entangled.
So it's a
down
balance, exactly. It's a balance. But yeah, we move on.
Okay,
Yeah,
we
yeah.
just saw out how built up.
Okay.
Yeah, that was a try, but we move on and have a last look at the little sponges. So after we're gone, the sediment will go down again relatively quickly. So yeah, matter of some minutes to an hour or so, probably. But since we're constantly staring with the propeller that the ROV has, the really fine powdery sediment up here, it's more tricky to sample in this area.
I'm taking off some of my downs, but as you can see, we're moving slightly. So I'm on borderline from we might be okay here. You can see anything?
Nah. No, not really.
Whee!
Okay, so now it's better because you actually...
I've reduced my downs, but again, movement there, if I come off it, I'll probably start lifting off the speed limit. So, getting borderline with how much I take off. Because obviously that's causing...
Yeah, but now, nah, it's okay. It's okay, we'll find out at that place.
There you go.
Clear enough to...
So we're looking a little bit more still at this area with interesting sponges on the lava rocks and discussing if we're moving further after all, or if there might still be a way, or how we go about for the next sites to make it easier to
Meaning that if now
to take a sample.
that you need to set again and things will start.
You tell us what you want, we pick it up.
But
we can pick it up quicker than...
yeah, but no, because in the other side there was a little rock, but not here.
On the right side, in front of the right arm, do you see anything interesting? That's the area that we can reach.
No, but those are giant rocks.
But we're going to have the same where we move to, or if I move forward, or it's going to disturb the seabed again. So we can have the same potential.
Yeah, the same
but you're going to
problem.
have all of everywhere down here.
I mean, this is what we want, but this is not even a rock. So this is
zoom out from that, so I can see where you are.
this is outcrop.
Yes,
so
unfortunately, we cannot take just a gigantic rock with us. So we have to look out a little bit until we find the right thing.
Right, sorry, you want a whole rock or something?
Exactly, so many things that has to happen at the same time.
Right, okay. No,
so it's okay. Thank you.
You got good fish now.
I think it's cool on the rock in the middle, for example, we still see some of the layers that the lava once had before it dried.
Okay, thanks. Yeah, actually now we moved again because we were
And a
going-
lot of sponges on it,
we were
the
opening
little
the-
ones.
Crossing the line here as well.
Sorry?
Going across the yellow line now, just for the line.
Okay.
If you see
Yeah,
that.
you can keep it with the line. Yeah.
Do you want me to follow the yellow line? Yes.
Yeah.
Thanks, yeah.
Yeah, we'll see the other crack. Then.
Ooh, lots of little sponges here.
Just bring the head down, up into 50, now we see what we like at that.
Oh, those are nice
we
encrusting
try to
sponges.
stay steady
Are
here
we-
for a while?
Can we stop for a while?
They look interesting, don't they? And some worms in between.
There must be also Assyrians. So this one in the center is an Assyrian, I think. The one that is a little bit more whitish. The new
More transparent.
The new species.
Oh, oh, there was a crinoid on top. Oh, there was there's a little cave or bridge of lava. Ah, more crinoids sitting over there,
Yeah.
just flowing in the
We
currents.
try to climb this to see how up it goes.
So we now climb the rock wall and
Finish. So
are
okay,
on top. So we
this
see a
one,
lot
for
of
instance, we would like to have.
see a nice sponge. Do you know what it is?
No, that's
Ooh,
why I
unknown
asked them if we can take a little piece, just a little piece, to see if it's
Just
a
a
new
little.
species or to see what it is.
Is it
That's
a fan?
Yeah, it's
A
fan shaped.
fan-shaped sponge. Yeah, sponges have a lot of different forms. Some of them were more like tubes, others like crusts, others
We
like
actually have frogs.
fans.
You see,
What?
Francie, we actually have frogs.
Oh,
Free
hello.
samples.
But I don't see any.
Free samples.
But I don't see any sponges on them. But I see different kinds of rock.
Have a
Like-
geologist on the vessel.
Yeah.
He will be happy.
Yeah, look, there's like the cracks were filled up with some other kind of rock composition afterwards.
The problem is we've got rocks underneath me,
Yeah.
and I'm trying to see where it's at. And we've got a big drop.
So we're interested in the sponge we've seen before that was quite fan-shaped, and we don't know what it is, and especially the sponge expert doesn't know what it is, so it could be something that is yet undescribed, and that's why we are trying to settle down now
Yeah, I want to see
and
where
try to
it is.
take a little piece of it to find out what it is, if it's new, how the spicules look like that we talked about before. So the little, the little glass spikes sponges usually create, and by looking at those under the microscopes at these little spicules, we can, the sponge experts can tell which species it is because they're very distinct.
A sponge.
Yeah, none is like the other. I sometimes think of it also as like snowflakes or so, when none is like the other. But yeah, if you see the different forms that these spicules can take, it's really amazing, I think.
Problem is,
What,
well,
yeah, what just
Maybe
nature,
it's
so to
next
say,
time before then we can
created
try to- with this
there.
one to see where it
Yeah. Yeah, we can. Yeah, we can. Yeah, try to handle that, but we need to see where this goes.
But to get this, we have to create this
We got a
little
canyon with
sandstorm
rocks that
again.
just can't go there. Just put down. So we keep getting over these rocks. Plus we've got some rocks there because when we come down.
And we need to discuss quite in detail what is a good position for the ROV to land, because we sometimes have we have rocks around us and need to pay attention to these. Yeah, we don't want to.
But we're coming up, so I need
yeah.
to go around the other
We
side.
don't want to damage
Explore
the ROV or tilt over or something like that. So we just need to see what works best, and also how we could potentially sample this. And if we did sample a little piece of that sponge, sponges are very, very good at regenerating their body parts. They can even, from just a bunch of few cells or even single cells, regenerate their whole body. So yeah, they're pretty cool in that regard, and in other regards, I guess, as well, as Julio would probably tell you.
Yeah, we won't see it again. Let's just keep moving.
And little shrimp, I guess.
And then it's over there.
Sorry. Can you straighten up your camera? So then I can
you mean press?
Yeah, because I have no vis, this is looking down.
Yeah. Okay, so
I've got a watch that I don't hit any rocks.
Okay.
Okay. So if you turn
that camera
straight, no, put it straight. The HD. So
We put the immersive camera back. Just have to think of it before we take the sample. No, if we would be ready to take a sample, we put it back in. But I think everyone has it on. Sorry?
Now it's recording the camera, special camera.
Ah. So we are still trying to land at the moment?
We're trying to see where it is.
Trying to see where it is.
We're to see where it is. I can't land blind.
No, better not. Yeah, it's a tricky area.
I think that we can
Yeah, so since there are rocks underneath us and we cannot really see them at the moment, and we have to regain our orientation, we just have to be extra careful in this dive with the sampling that takes place in our navigation.
You can have a look with your camera, and then when you spot it,
it's just that I can't see nothing. So. So honestly, I think it's better to keep moving.
the thing is, because we're in a canyon and I was on the top, I just can't go in.
Yeah.
Because if I go in the canyon,
Yeah.
I can't swim. That's just game over.
Yeah.
You know, if we get caught, there won't be any more. So it's game over.
No worries.
Yeah, the characteristics here of the bottom where we are right now are quite tricky because it's like a little canyon and we cannot go in there with the ROV because then it's like hard to get out of again when we're surrounded then by the
just got
rocks.
steps on it.
Yeah.
The wall we we had vision something, but when we were close, we just lost like this. But there's a canyon down below us, and it's just over the precipice, just over the precipice of the canyon. We've come forward a bit there now.
And we're at the moment at the handover for the ROV shift. So one shift is handing over to the other, so that's also why we have more people here around. And the rock pilots that we had before, they're going on their well-deserved time off. They work in 12-hour shifts.
Be careful
as
because
we
tech, it's not very exact heading of the
yeah, from 12 to 12.
We were converging to the
So some of them now have their well-deserved end of working day, and the others are just starting it.
Yeah, if we find something interesting, we may deviate a little bit from the line. Yeah. But otherwise, we follow the line.
And just thrust them back a bit because the current's taking us along that line. People might want to jump in.
Thank you.
Hello.
Hiya.
Hi, how are you?
Here we go again.
Yes, Rob taking over now.
Sorry guys.
Huh?
It's,
all good.
Oh, no,
Thanks. Thanks a lot.
I'm going to stick in the canyon.
Always nice.
Oh yeah, no.
Possible.
That's great.
Thanks.
Thank you.
Cheers.
Cheers.
Okay, we keep moving.
So we continue our search for but it's because we have very, very powdery, fine sediment here that is very easily distributed in the water column, not dissolved, but suspended. Suspended is the word, right? Yes.
Resuspended.
But yeah, after we leave, it will also easily settle down again, because we're just above it actually, and it's already piling up like this, or suspending like this. Yeah, but of course, like this, we cannot see anything, and that's not very good.
Good afternoon.
Julio, while you were navigating and discussing, I was telling, like, for the case that we would sample a little piece of sponge, that sponges are really great at regenerating their body parts.
Yes, that piece will regenerate without causing major damage to the sponge. That's the advantage of being of not having true tissues, meaning that every part of the body can regenerate very easily. Yeah.
So even from like one cell, some cells, they can just grow all again.
Exactly.
Whole again.
Cells are not that specialized in the sense that, for instance, we humans, our cells, when they're specialized, they cannot go back and they cannot switch between type of cells. So a brain cell is always a brain cell. But the sponges, they have a little bit more of flexibility in that sense, because they are more, let's say, not simple, but the tissue-wise, it's a little bit more simple. So that has many advantages. So yeah, you can break a sponge in many pieces, and the pieces will regenerate. So you can make clones. Actually, you take a sponge, you cut it in parts, and then you put that parts in different parts of the sea bottom, and they will create a different sponge. Every one of the parts, they will create clones with the same genetic information.
Good afternoon again.
That's pretty cool.
That's why we try to, when we see a large sponge, we try to collect just a piece.
And then we know this is not the end of the sponge. It's not hurting very bad.
Thank you.
And like having a wound forever or so, but it's just gonna be like, oh yeah, I'm gonna grow that back, I guess.
And for us, that's more than enough. We can check the skeletal elements, so the morphology. We can check the DNA. We can even check if there are secondary metabolites, so interesting chemistry, with just a piece of the sponge without killing the individual.
And isn't it also that these secondary metabolites, they can even be used also in pharmaceutical ways?
Yeah.
So they can also be used for, because the sponges have it to also have some forms of defense mechanism against bacteria.
So they cannot run away. So that's the way they protect themselves, having these molecules. And you can use those molecules for many different things, like against cancer, against as an antibacterial, drugs, as sometimes some of these molecules are very nice to avoid things to settle on the wood of the ships and the vessels. So there are many applications. So we always say that the biological diversity of sponges is in parallel of chemical diversity. So this is also a research that sponges have that we want to protect, not only for the environment but also for ourselves, because we can learn from these molecules and produce new drugs that could potentially save lives and be used in the biopharmaceutical market. Yeah,
there was an amygdalus, no?
I think so.
The one with the yellow.
This is a little endorix, but I am a little bit afraid to stop. I'm really.
The ground is so soft, guys. The sediment is just going to go everywhere.
Yeah.
We're just trying our best there.
Yeah.
Come down a little bit.
And we saw interesting little mounds in the soft sediment, I think.
It's over there.
So also an indication that something's living there underneath. Oh, and sponges coming up in the distance. On the right, what was it, do you think?
There's a big mound coming up, about 25
Fish.
meters high.
Big rock.
Yeah, right.
We have bigger rocks coming
Mound.
out.
There's a fish.
Mhm. And another one there. You zoom in a little bit. Ah. Definitely a fish.
And we have
And the
this
little-
field of
yeah.
stems.
Huh. Jelly?
Yeah, kind of a jellyfish.
Very dark jelly. What are the more pinkish? No, it's just one. It's
It's a shrimp.
Ah, shrimp. Yeah. That makes sense. Oh, a lot of them. But they are bigger than the ones that we've seen before, it seems to me. Or maybe we're just closer.
So now this.
Is it Amygdalus?
Yes.
Acopherus.
Acopherus, exactly.
And Lysodendoris.
And there is another.
Our spaghetti sponge. Is it really a common name for this, or did someone just make from us make it up? Or
It has no common name yet. We need to think about one.
spaghetti name is spaghetti sponge seems suitable for me.
Spaghetti sponge.
And those patches?
Yeah, that's
Encrusting, or
that's the same community of encrusting sponges as
Yeah.
before,
and the shrimp.
and the shrimp.
Many shrimpies. Fishies.
So there you have an asconema.
Oh, you can tell that.
You see the big one in the
Mega
that is like
Atrialia, no?
potential, yes. And here, this is the community that we are looking for. So the new species is the more like orangish. You see?
Yeah. Like on top there. There. Or on the left.
But you also have little Emigilius, so they are also like kind of oranges. So this, for instance, is not.
Little the Amygdalus acopherus.
Yeah, the little ones look like that as well. So that's why we really want to stop and have a close up.
We can sit on top of the rock.
Yeah,
Yeah.
that would be perfect.
Ooh.
Okay, so stop. Stop. It's 11:05:07.
Yeah.
Okay. So now we're stopping on a rocky outcrop. Maybe will be better. Maybe we won't have so much mud this time.
So the current is up the current is coming from the starboard side. And it's clearing out, so
Great.
Yeah.
well, as the current is coming from the starboard side, we could always spin the head round so it's coming into the current is coming into the vehicle. So if there's any disturbance,
We're discussing some strategies what could make it easier for our little sandstorm to settle,
So I
or
have a
for
tendency
view to
ask a
clear.
lot, but if you are not okay with stopping, you just stop.
Yeah.
No, don't, because I don't know all these things. So.
if you want them to stop, I'll say yay or nay, as simple as that.
Exactly.
So the little Amigellos we saw, those are still baby ones then, so to say?
Yeah. Yeah, small specimens.
We've already seen some that are now in their reproductive period, no? So that we've seen some sponges that have
The Lysodendorids,
butts
yes.
or even eggs inside.
All right, yeah. And Sergi was checking the Lysodendorids, which is the species we are targeting for our connectivity study, and some have eggs, and others, I think, he saw sperm. So that's very interesting, meaning that something's going on. Maybe now is the time they are reproducing. So, yeah. All these things will be processed on land and it will take our time. But yeah, it's very promising.
And we see also the different ways how sponge reproduce. Some of them, they just clone themselves, or they like bud and then they have a new sponge falling off. And others are like broadcasters, so to say. So some are females and some are males, and then they release their sperms and eggs and hope that they find each other in the water, and then create new little sponges to settle on a suitable substrate, I guess.
Budding is widespread in sponges, and is a way to produce clones that is a little piece of yourself that will float into the water and try to colonize a different place. But then you lose the advantages of sexual reproduction, which are becoming adaptable to the changes of the environment.
There are more mixes in the gene flow.
Exactly. So yeah, it's a balance.
So you would say more.
I don't think it's going to clear.
No, because I think that is.
We'll
because we are
learn.
extracting-
I'll ask him more about sponge reproduction when he has more time, I think. But yeah, some sponges are hermaphrodites, so they are both male and female, or neither in one. And some are, they have distinct sexes. They are male and female separately.
Okay, we move on because it's not going to clean.
Yep.
There's more rock coming up. When you see the white, they're the rocks.
Oh yeah.
Hard contact.
Perfect. So maybe we can try that to face the current.
So we're moving further on our hydrothermal vent area, field area. So this old volcanic area where we can see some of the lava rocks.
Yeah, there you go. finding those rocks would be perfect for us.
The little ones, right?
Because they're small ones.
So we want to sample little rocks because we've.
So I thought there was always one
got a crinoid. Why do we want to sample little rocks?
Well, because we are looking for this new species that appears the last day, and it grows into these rocks. So.
And we want to do genetic analysis so we can find out if it's indeed an undescribed species and about its characteristics, about their structures.
It's okay.
And yeah, they grow on these rocks here, and we want to sample a little bit.
Another day we'll find a similar community, so I'm happy with that. We'll be less mad, so no worries.
So I think now we're moving a little bit over sediment area, and then we find some rocks again.
Yeah, but I think we will not stop because it's every time we stop the mud resuspend and it's pointless.
So close yet so far. How can I tell the baby Amygdalus aquifera from the potential new species, undescribed species?
That Amygdalus, they produce, well, I think that are buds, like little projections on their skin, while Amygdalus is smooth on the skin level. When you have them on your hand, they are very different because Amygdalus, the new species, has a lot of mucus.
Yeah.
While the
so
other
they're
one,
very slimy.
Yeah, super slimy.
Yeah. Oh, right, yeah.
And I think we're almost at the end of our second transect here in this hydrothermal field area.
Yeah. Interesting.
And we see another rocky valley here with the sponges positioning themselves nice in the currents. There are not so many carnivorous sponges out here, are there?
I've seen some, huh?
But less than before, seems to me.
Less than in the coral reef, for sure. Coral reef was something spectacular. I've seen that before.
So many carnivorous sponges particularly, you mean?
Many, yeah.
And yeah, that was about 1,000 meters depth, so now we're much deeper. Is that because carnivorous sponges are more?
No, I think it has to do with the food availability. So in the coral reef there was a lot of marine snow and a lot of current, more than here, I would say.
Ooh, a lot of crinoids there.
So,
It's just too violent. Yeah.
I'll try to get those crinoids. It's tricky with the moment. Goodbye.
Yeah, all hanging out there. And a little jelly. How are we going about with the end of this transect? Oh, sorry, did you?
Yeah, I think that we are good to stop the transect.
Yeah.
So it's over
because, well, oh, okay, we have a Lysodendrix, that's, no? Where?
Yeah, it was to the right.
sorry?
Because we're almost at the end, or like parallel to the end of our transect.
When they're
We were
turning up,
wondering.
when they go north.
Yeah. Then we'll mark also that it's
Yeah.
the end of our transect here, and then we can start moving
We turn
upward.
there now.
Yeah, turned. Yeah.
Cool. Thank you.
Right.
End of transect
Yeah, end of transit.
2.
Okay, so now we will keep going parallel to the northeast. Yeah, yeah.
It's the change of vessel.
Thanks.
Not
Yeah.
yet.
Do you have
Reach our feet.
the folder
Debrief.
with the profiles?
Can you change the bearing slowly?
Because we have a nice folder where we printed out all our little, our different transects and their profiles, how much depth, gradients they have. Yeah, so now we're moving. So now we're gonna move northeast for about 300 meters, and there'll be more valleys and crevices coming up. But I think we'll have some time going over a rather sedimented area before that. Yeah, we're gonna be at about 2,700 meters depth. What's
We consider that we have started the transit trip, or do you prefer us to go a little bit more to the north?
I think going to the north looks interesting, you know?
Yeah, we are going north, but are we going to the line, or we are just going in parallel to the
I
line?
would say we're
In that
going.
case, we will go like this.
Yeah, I would say like that rather parallel to the line, because it looks similar if
Yeah,
we're going parallel.
I agree. Yeah.
Yeah.
So we have started transit trip.
Yeah.
Great. Transit trip.
Where do I put our paperwork?
That's okay.
Underneath. We hide it underneath.
Play with the fields.
Ooh, is there this sponge coming up? Julio, sorry. But is there this sponge that whose name I forgot?
Asconema.
But the Mega Australiana line. It's so at each end and start of the transect, we diligently write down the coordinates and the depth to keep track of everything.
That's it. That's it.
Oh, jelly to the left.
Mm-hmm.
Or, yeah,
Yeah.
jellyfish. And we're moving onward on this yet unexplored deep seabed field of volcanic area. So this area has been mapped before, so we know the topography, but no one has ever visually captured this before, been down here with a camera.
Yeah, it was a little bit of blue. We were on the top of a little mount.
Mm-hmm.
So now we won't see anything for a while, maybe.
you go up a little bit? Why? Because there was a gap.
Yeah, so we're currently at about 2,670 meters deep. On top of a hill, and so we're moving a bit until we can see that again. Like
Yeah, hold on, because they had to fit something with the cable on
Yeah.
Barobi.
okay.
Don't worry. There is these things.
Yeah, just stop a little bit because we have some gap here on the drum. So we need to pay out a little bit and level this so we can do threads even.
Okay. Yeah, on the ship here we have a big drum of a very, very long cable of 4,000 meters, I think. 4,000 meters of cable on a drum. And we have to make sure that there's not a big cable salad. Do you say that in English, do you think? Do you say that in Spanish? A cable salad.
Salad.
A salad of cables, so when all your cables are really messed up, we saw it's cable salad
I don't
in
know
German.
if we have an expression for that.
I've never heard that in English, I think.
I don't know. Leo, I don't know.
Let me out.
Yeah.
Okay. Go.
So we want to avoid that from happening, so
Cable salad.
yes, so we're fiddling here.
How you say it in German?
A Kabelsalat,
Cable salad.
yes. So it's actually very similar to the English word.
Well, at the same time.
Makes sense.
Yeah. When I was a child, that was actually a big thing because we had these cassettes for audiobooks, music or so, and when something was wrong with the cassette recorder, it could create a big cable salad, and then your cassette was messed up, and you needed. Do you remember that as well? You needed to roll that, but you didn't have a name for the mess created. No.
Not that I'm aware of. Un lío,
Yeah, it was
de
a nightmare.
cables. Un lío de.
You never knew if your cassette worked afterwards or not. So it was too badly injured.
Yeah.
But yeah, let's move back to the seabed.
Just come down 30 meters.
Okay.
Okay, all stopped?
No, keep going.
So they need to fit something. So now they're kind of steady.
All stopped?
Yeah, back up. If you need to.
Yeah. I'm okay here.
Yeah, we see on the soft sediment that there are some
We're quite way.
little bulbs, little mounds. We don't really see what is living in the sediment, on the sediment here, but we know that there's more life in there, even though it doesn't look that spectacular, at least to me. Yeah. And we're moving on to find more rocky areas with more sponges, and hello little jellyfish. Ooh, it's making little spins for us. And we have some more, I don't know, not holes, but probably these little stalked animals. Yeah. Yeah.
Yeah. Yeah, I wonder what are those.
But at first glance, I sometimes think it's like a hole because of the shading, but then no, it's also the stalked animal.
I mean, it could be many things. It's a
Well,
hole there to the left. To the left.
Yeah. There you can really see how much sediment is on those rocks and that there are really rocks underneath you, but very well hidden.
Because we're on a valley, we're on the rift.
So all the sediment
end up here.
concentrates.
Yeah, ages and ages of sediment falling, marine snow.
Yeah. Like a snowy landscape around here.
Yeah. These are androids.
Oh,
You see?
small ones, yes. We have small Isodendrix complicata. What's that?
The black thing.
Rock?
That's a rock.
Thing,
I think.
yeah.
Yeah, that's just a rock. That's probably the rock beneath.
Hmm.
Just taking this
I'm
little
not
bit
going
of
to
slack.
What was that? It's kind of
Oh, it's alright.
Come up 1 metre, so not really, but like 1 metre.
There are a lot of these, of the little jellies. Oh, it's the same one following us.
Maybe we can speed up a little bit.
Yeah, well, we've got just all sand in front of us at the moment. All sand.
It's just sand, right?
Yeah.
So yeah, we can speed up. If that's fine.
That's no problem.
We're gonna speed up a little bit. Oop.
Got
Not
rock.
missing to block the jellyfish. And
Let's see how they look like.
yeah,
because if they are
gonna
like
speed up
ones, so
because there's more
no chance.
sandy bottom coming up in front of us,
Maybe that now it's
and we're more interested
That ship's doing
in the
0.3,
rocky areas to see more of the sponges and other organisms.
Maybe you can come inside for now.
Okay.
So now we are a little bit faster because it's kind of sedimentary with everything repeating and repeating. So
But later there's more of the rift surface coming up.
in about 30 meters we have a interesting outcrop.
Reach RV.
Bridge.
Can we change the bearing slowly north, please? Still the same speed, continuously.
Changing to north.
So here we have more of our lava rocks. Ctenophora. Nice and sparkly. Oh, hey. So we can see the combs, the little combs on it that are sparkling.
Luminescence.
But we don't see it anymore, and I will lock it. And as I learned before, these jelly and the sponges have a fight going on, right?
Well, in the scientific community, there is this conversation going on about who is older, if Ctenophora or sponges. And there is every once in a while a paper published saying that it's Ctenophora or sponges. I'm not following this day by day, so I think that now sponges are winning for now, until new evidence for Ctenophora or sponges. So, but it's interesting. I would say that the sponges, that it makes more sense that the sponges are older, because the way they are shaped. So it's more easy to me, the evolution to produce a sponge than to produce a Ctenophora. Ctenophora, they have symmetry, the tissues are a little bit more specialized, but yeah, it's an interesting debate and conversation, or fight if you want.
And if we're talking about the time spans of age, it's like they're 600 million years or something old?
It's also a debate.
Yeah.
I mean, the oldest fossils that we know for sponges are at the Cambrian, around 540 million years ago. But then we have evidence that the sponges can be found in older rocks, just that we don't have the fossils. We have chemical molecules that we found on rocks that are very old, and in those rocks we find the same chemicals that are only produced by sponges. So this is one evidence that sponges are actually older than 540 million years ago. Yeah, those are sterols, the molecules that are telling us that probably sponges appear way before than the fossil record is telling us. Back to 600 or even older, 600 million years, or even before.
Yeah, unimaginable timescales. Far before any dinosaurs, before trees, before any other animal.
There's cracks in the rock. What's that? It's watchable. That's Asconema. Yeah.
The Mega Australia?
False alarma. Yeah.
False alarma. Ah, yeah. We haven't seen the other Asconema species at all at this depth.
Because we are too deep. Yeah.
So they are known to be higher.
Camera number 4. Here, but camera number 4 at the bottom.
Say it again.
We're going down. Let's look here and down. Flying over. No.
Over the crack.
If you look here,
Yeah.
this is underneath us.
Yeah.
We're looking down.
Okay. So it's a big crack. Big crack. Yeah. Is that problematic for you? Not for the gate.
I'm trying to look down, so I was telling you if you look with your eyes on that way, it's
Thanks.
Of interest.
yeah.
Next to the camera that we have mainly here in the screen in front of us, the ROV also has other cameras for navigation all around to be able to see what's below, what's above, what's at the sides, and to ease navigation. But also it's interesting sometimes to see what's actually at the moment underneath us, or what's next to us, or above, etc. So we sometimes have to be looking to the other screens as well.
Fish.
Yep. Oh no, I put the numbers on the keyboard again. Ah. Is there the Sedendorix coming up?
I don't think so. I think it's the Psychocalids.
What? It has a stem, no?
Yeah, that's Actinella. It's the largest Actinella. I've seen a couple of those.
What are they called again?
Psychocalids, I think.
Psychocalids?
Yeah, we want to double
yes.
You want to double check that.
Okay.
Yeah,
These
that one.
Psychocalids. And they also probably have a stem so that they can better filter.
Exactly. Those are
the
strategies to
water
sedimentary areas.
And not get so much sediment in their pores. But now for, say, because we're going above them, we're stirring up a little sediment, they can easily expulse that again, so it's not like really bad. Is there fish? Yeah. Yeah, it would be more problematic.
You see the carnivore sponges there?
No. Where's the carnivore sponge?
Oh, no. It was a false alarm. Sorry.
Yeah, it was a trick question here. Not with me. But there we have some sponges.
Yeah.
The stem sponges again. Is it the Psychocalids?
Yes.
Ooh.
Yes.
Jeremy. Can we change the bearing slowly again to 045, please?
045.
Thank you.
Ooh. We're already getting closer to the end of line.
Yeah. Have the transits here.
No, you have.
I mean, there's plenty of Psychocalids. So there's another one over there.
Yeah, I already used it. You're checking for the next one?
It will be the last one in this area.
But we have where do we have the overview?
I don't know.
We have another.
Yeah, sorry, I was not thinking.
No, it was correct.
This one?
Yeah, it was these three, but then we have another few coming up where we could do a blue transit, but it would take quite a while.
So now we don't have another one there. We need to do transiting.
But it would be a few kilometers, so I think we need to check if we indeed want to do that or not. Yes. Maybe I can check.
Could we follow the crest?
Crest?
Yeah.
Your current is going to just start opening up. Even if
really bad viz coming across that. Is that what you want to do?
Well, we want to get to the point, but maybe instead of going
Yeah, we just want to have bad video.
It's not going to go well in that direction because of the current.
Oh, okay,
Yeah.
that's fine. Just to go to the point.
Yeah. No, it's fine. Yeah. So we should check if because we end this transect here normally, how we'll go about with the next transects that we planned. Shall I go up and check with them?
We still have some meters to go, but yeah, you can go and ask maybe.
I think they want to come back, and
Sorry?
they're debating if they're going to come back. Sandra.
Come
she's gonna
back where?
She's going to come back to tell us.
Oh, they're already discussing. Good. Thanks.
So that's the listen and Doritz, you said?
So we can just hang out a bit here. Yes.
So, yeah. No way to collect that one, huh? Vale.
No viz.
I ask, just in case.
Yeah, we would like to collect these sponges called Lisodendrix complicata because we try to find out more about their community structure, their connectivity, so how they're connected between different locations, also from their genetics. Right?
So yeah, genetic connectivity is important because then you know how connected they are and how resilient they are. So if you have a population that is very isolated, then that rings a bell. The more connected they are, the more resilient they are to whatever is happening, to whatever disturbance may happen.
Amygdalus.
That's amygdalus.
And videotape change in 10 minutes. 10 minutes still.
Yeah, that was amygdalus.
Acropherus.
Acropherus, yeah. And this community is just repeating and repeating and repeating, so...
Yeah, go for it.
I wonder how many species are there. Yeah, I would say that
all these dots.
Maybe 20.
20 of the,
Sponges.
like the little dots there.
Something like that.
And none of them is described yet? Like potentially?
No, I don't know. No,
yeah, we don't know.
I
I'm
don't
always
know.
just potentially speaking. Or what we expect at the moment.
We ring a bell in one because it's a genus that there are not so many species and they are usually shallow water. So that rings us a bell, and that's why we say that probably it's a new species. And that's the Lisodendrix. But for the rest, there may be, of course, many new species, but it's a little bit more tricky. If you have a group that has so many species, you have to check all of them just to know what you have. So that's time-consuming. It's not something you can do it so quickly. And often you have to go to the museums and compare to the material that has been previously collected. Sometimes you even have to sequence that material and compare with your sequences of your new material, and this is a process that can take years. But yeah, we will work on that.
Also small Lisodendrix. Also.
What's that?
Looks
the
like
one,
a telephone.
That's the one I want. That's the one I want. That's
Is
the one.
that?
Yeah, that's the one I was trying to collect before.
The amygdalus?
No. This I don't know what it is.
Ooh.
This I don't know what it is.
Come up, just try not to disturb the disc too much and let that settle.
He's just going to hang and wait so you get a better view of it, okay?
Perfect.
Just going for bridge for 1 second, okay? He's in 9 minutes. So don't pick him anything too much.
Don't think they're going to get it. See how it's drifting right over the... Do you see how it's drifted right over where you want to go?
Okay. Where did it from? Okay.
Well, we're 7 meters, 6 meters up.
So now you're trying to see what's going on with the clouds.
Yeah. So it's gradually going that way, but
it seems like the current goes.
But the current's coming from behind us. So as we disturb it, we're going faster than what that is. But as soon as we stop, that comes
It
faster
comes,
than
it
what
catches us.
catches. Yeah.
Is there a lot of...
So we're discussing at the moment
No current.
current,
what
that's the
the
problem.
current is also doing to
So if we
our
went
visibility
that way,
and the sediment and how we move, because at the
the
moment
disturbance would be behind
it
us.
comes from behind the current,
Well, then
so
we head south.
it
We want to head
flows
north.
past us from behind, and yeah, we might reconsider the bearing.
No, I guess that the reason why there's so much mud is because there's no power in the area.
Basically,
It's always,
although it's soft. Yeah. Like I said, if we're coming down, if we go north to south, the current is going south to north. That's the perfect combination.
Yeah.
But we're not.
Yeah, no.
We're going north, so's the current.
You have the wind from the tail.
Yeah.
So we are running out of multi-unit.
To the unknown.
Oh, now it's
We
moving
really are going to
completely into the unknown. How exciting!
Yeah, no, just
Our map of
Just follow-
bathymetry
just
data
follow
is
the
also
full line.
ending. So
that's the problem.
if we move further in this direction, so we wouldn't have a clue anymore about the topography of the seabed.
I mean, 2 days ago,
which
the
would
community was
be
very
very
similar and it
adventurous,
was super
because
clean.
normally
I
you
don't know if were
always
there.
you want to have an
Not-
indication
not
about
But that's
what is the
thing, we're
profile,
always heading
like
to
where
a
are you
current.
heading? Because with the ROV we're
This
mostly
occasion, we're heading
trying to
with
go
current,
upslope
so
and
good.
not, for example, downslope,
But in any case,
and
what
to
I'm
not
looking
disturb
for, it
any
will appear again
equipment.
Yeah.
during the
We
cruise,
try
so
to be
it's not
always
not the end of
cautious
the
of
world.
what's coming next.
Okay,
That's fine.
that's manual, okay?
Okay.
I'm just collecting now, paying in, because we are now stopped. The ship is coming.
Oh, and tape change in
Going
5
slowly.
minutes, I'm reminded here. So tape change means that the recording of the video cartridge will end in 5 minutes, because we have set 2-hour tapes. So after every 2 hours we do a backup of the video that we have recorded.
Yeah.
So in case there would be an error, a corrupted file, we wouldn't lose too much of it. So we always have like a safety net. So never more than 2 hours of tape can be lost. And it's also a way of like switching around here a little bit and yeah, bringing some shift changes. What a nice transition. Here is Anna-Helena ready to take over soon. How are you doing?
Oh, good. Just come from lunch and you can look forward.
Yeah.
Very
How was
different.
lunch or dinner actually, if we went by
Our timing?
our timing?
It was good. It's always good food on board here.
Indeed.
That's wonderful, right? We are lucky indeed.
Yeah, we're being served very well.
Have you made any samples?
No, we have not
Yeah, it's
succeeded
impossible, right?
in sampling, yeah, without creating.
Did you do like we
sensing.
tried to do, to sample the earth?
No, we didn't get that far.
Actually, we got a rod. Yeah.
Ooh.
Yeah, yeah, true. We did some.
But it's impossible. He was trying to
accidental
face the current. And now I'm not sure if we are standing by waiting for Sandra's instructions, because this is the end of the trip. Do we have what to do now? We should do transit if we go out.
Yeah.
No, no se puede.
So are we at the end of
Yeah, yeah, we're now really going into the unknown at the moment. Yes, we followed the second transect parallel to our usual line, and then we went upwards and went also a little bit, yeah, also parallel to that line, and discovered more of those areas. And we've also seen a lot of these communities of potentially unknown sponges. So it was quite hard to always see them and not be able to collect them. Also, yeah, also viele Sudenderex.
$2 minutes until we...
Yes. 2 minutes, and then we'll change the tape, and then we make a backup recording, a backup of the recording. And yeah.
A ver, las gestas esas más o menos coinciden. Y el bonito todavía no es una fractura, porque eran agujeros muy grandes, pero bueno, vida había en esa época. Creo que decía Agustina que
And for the sample you have taken, where you went quite into a bit into the unknown, we've seen that you've collected a rock, but we're not sure yet
What is on the rock?
What is on the rock.
No, it
that
was
would be a nice
invisible.
surprise.
So that
Yeah.
is, yes, sometimes we need a surprise.
Yeah,
right?
yeah. We want
It was in
surprises
the
in our
Video
lives.
in 1 minute.
on the other side.
Oh, thanks.
Yeah, you know, if you don't go for the unknown, then you will never learn new things.
Yeah, that's the spirit.
Había una de esos.
Exploration instead of always exploitation.
Seguro, no era emisiones. Ah, bueno. Claro, eso te has fijado que hay una handshake pero blanca. Esta es la que nos falta. De la comunidad. De resto saldrá.
How far? Huh?
You're on your way to the next one.
28 seconds. 27.
So,
You're waiting for the decision.
I would just write end of the transit, right? We agree on that. Yeah. Okay, so 11:56, 55, yeah, transit. That's two. So this is
Completely. So, like, we're aiming
all right.
600 millimeters off the
Let's
sea.
change the tape now.
Oh yes. Can we change the tape?
Yeah.
Stop recording.
Okay. So it's just that's life.
Stop.
One,
Yes. 2, 3.
These steps allow us to compare like a shallower versus deeper animals. Adaptation, right? We see that we're gonna sink right there. Yeah, that's the problem. It's so light. No problem. We can do a water sample once we finish the dive. We'll set up again just like we were.
It would be nice to test that because
Big time. It's on my mind. Yep.
I can imagine that.
Okay, see you.
Good to record again.
Your lunch,
Yeah.
dinner.
See you. See
I
you.
have 2 formats. This one, okay?
So we're putting a new cartridge in there now, and also indicating in our logging system that we changed the tape, so we will know afterwards what is on which tape and when was it changed. Are we recording again? Are we recording again as the tape changed? Okay, okay. Yes.
So, are you ready?
I'm ready.
1,
Yes.
2, 3.
Thank you. Yes. And start recording again. Thanks. All right. Thanks a lot.
Enjoy lunch, dinner.
Yes.
And here is the cartridge.
Oh, yes. I will take that for backing up.
So this is Anna Helena and Sergi being back after a little bit of a break. And and we will continue. Ooh, there is a crack of some sort.
Yes.
So who knows what that is? But it's an exciting little crack there.
Back. Yes.
So, did you have a good break, Sergi?
Yeah.
Yeah. I have cleaned the lab.
Long enough, it's fine.
Yeah. I spent the time cleaning up in the lab. So, of course, when an ROV dive comes, the ROV come back to the surface. If you have sampled a lot or some at all, when it comes up, we have to preserve those samples and make sure that they are being taken care of so that we can use them well. And so we have spent most of the time since last evening preserving the samples we got up.
Yeah.
It's not just pouring them into something. You have to make sure that you are sorting through all the different little milliliters of water to check if there is something in the water, and to make sure that we note all the details about where they come from. And so it takes a lot of time to, and then get them into the right fixative, right?
For the different purposes.
Yes.
Yeah, so everything that is being taken as a sample, it's being used on deck. And we are 3 people in the sponge team, 3 people in the station team, and they're like fully devoted to preserve on time, because it needs to be done as quickly as possible.
Yes, especially if you want to look at the DNA. So I got up at 2 in the morning to start sorting samples.
Mm-hmm. That's what we are here for, right?
Yeah, that's why we do. That's why we have shifts. So there's always somebody awake to work on whatever is needed to work on. I think these cracks are interesting.
Let's just have a look. Oh, still a little bit dirty.
Because we're still in a very soft sedimented area.
Yeah.
And then we have these cracks.
Yeah, cannot see super well here. It's a little bit.
It's a little bit sandstormy.
Yeah, again. We thought we got rid of this, but
They're going to start moving.
Yeah.
Yes.
Sure.
So we will have a blue transit.
A little bit.
Yes. We are moving to a new transect, and we will lift off a little bit of the seafloor. Even though it was a nice little crack there, we will just go a little bit up from the seafloor so that we can move without swimming around, flying around in a sand cloud. And so we have a new
Okay, troops.
location.
That's this dive finished. We have 3 kilometers to the next station. And there's too much, so they're saying we recover the surface.
Okay.
Okay.
So we changed that. We go end of the dive and we will go to the surface now. And that was a very quick time in the shack for us.
I will just make sure
But
turn off upstairs before we come off.
Yeah.
All right,
we will hang out a little bit here and start
Can we stop the movement, please?
start with the finishing of this
Thank you.
of this dive.
Yeah. Was it
And
time?
I am sure we're going to get to new live streams soon.
With that,
So follow from both the international landing page or on social media to find out when we are on again. This of course always depends a little bit about weather and how long it takes to go to the next place we will dive, and the decisions that are being made here on board with the captain and the leader for the RV team and the leaders for the expedition on where we're going next. So while we're waiting to go up, we can look at this strange crack down at the right-hand corner. It's kind of Cool, huh?